Close your eyes and imagine stepping into a world where every surface pulses with life, where the very air you breathe carries the essence of countless organisms working in harmony. This is the rainforest, Earth's most complex and nurturing ecosystem, a living cathedral where towering trees form the vaulted ceiling and every layer tells a story of adaptation, cooperation, and survival. Tonight, we'll journey through this verdant realm, exploring how the rainforest doesn't merely house life, but actively nurtures it, creating conditions so perfect for biological diversity that scientists are still discovering new species hidden in its emerald embrace. Standing at the edge of a tropical rainforest, one's first impression might be of impenetrable green chaos. Vines tangle overhead, broad leaves obscure the view, and the density of vegetation seems almost overwhelming. Yet beneath this apparent disorder lies one of nature's most sophisticated architectural designs, a vertical metropolis that has evolved over millions of years to maximize life in 3 dimensions. The rainforest is not simply a collection of trees growing together, but rather a carefully structured community where every level serves a distinct purpose in nurturing the incredible diversity of life that calls it home. The architecture of the rainforest can be understood as a series of distinct layers, each with its own characteristics, inhabitants, and ecological functions. This vertical stratification creates what ecologists refer to as microhabitats, specialized environments within the larger ecosystem that provide unique conditions for different forms of life. Just as a city's skyscrapers house different businesses and residents on each floor, the rainforest's layers accommodate species with varying needs and adaptations. At the very bottom of this living skyscraper lies the forest floor, a world of perpetual twilight where only about 2% of the sunlight that strikes the canopy above ever reaches the ground. The floor is carpeted with a thick layer of decomposing leaves, fallen branches, and organic debris that forms a rich, spongy substrate. This leaf litter, constantly replenished from above, creates an environment that remains consistently moist and relatively cool, typically maintaining temperatures between 20 and 25 degrees Celsius throughout the year. The dim conditions of the forest floor might seem inhospitable to plant life, yet this layer teems with organisms perfectly adapted to low-light conditions. Fungi flourish here, their networks of thread-like hyphae spreading through the decomposing matter and forming crucial partnerships with plant roots. These mycorrhizal associations are fundamental to how the forest nurtures plant life, with the fungi extending a tree's root system many times over, helping it access nutrients and water from a much larger area than would otherwise be possible. Large-leaved herbs and ferns dominate the sparse plant life on the forest floor, their broad surfaces maximized to capture whatever filtered light penetrates the canopy above. Many of these plants have evolved remarkable adaptations to thrive in low-light conditions. Their leaves often have a distinctive dark green coloration, packed with chlorophyll to squeeze every photon of energy from the dim environment. Some species have developed leaves with special cell structures called drip tips, pointed extensions that help shed the constant moisture that might otherwise encourage the growth of harmful fungi or bacteria. The forest floor serves as home to the rainforest's recyclers, the countless invertebrates that break down organic matter and return nutrients to the soil. Termites march in endless columns, processing wood and plant matter with the help of symbiotic bacteria in their guts. Millipedes, with their many legs and segmented bodies, work their way through leaf litter like living composting machines. Beetles of every conceivable shape and size scavenge, hunt, and decompose, while ants form complex societies that can include millions of individuals working together to process the forest's bounty of organic matter. Moving upward from the forest floor, we encounter the understory, a layer that extends roughly from 2 to 20 meters above the ground. Here, the light conditions improve slightly, though the understory still receives only about 5% of the sunlight available above the canopy. This modest increase in illumination allows for a greater diversity of plant life, including young trees reaching toward the canopy, shrubs adapted to filtered light, and a remarkable variety of climbing plants that use the existing vegetation as scaffolding to reach higher light levels. The understory is characterized by its relatively open structure compared to the dense canopy above. Trees and plants here must be patient, growing slowly in the dim conditions while waiting for their opportunity to reach the light. When a large tree falls, creating a gap in the canopy above, the understory plants suddenly find themselves bathed in direct sunlight. This triggers a race for the light, with vines, saplings, and shrubs all competing to fill the newly available space. Some plants spend decades in the understory shadows, growing imperceptibly until such an opportunity presents itself. This layer serves as a crucial nursery for many canopy trees, which begin their lives in the filtered light below, before eventually growing tall enough to join the upper levels of the forest. The understory's relatively stable conditions, protected from the extremes of weather by the canopy above, provide an ideal environment for young plants to establish themselves. The temperature here remains remarkably constant, fluctuating only a few degrees throughout the day and across seasons, while humidity levels stay consistently high. Many of the rainforest's most spectacular plants call the understory home. Large-leafed aroids spread their foliage like green umbrellas, while delicate ferns create intricate patterns in the filtered light. Palms, with their distinctive fronds, add architectural elegance to this middle layer. The understory is also where many of the rainforest's most colorful flowers bloom, taking advantage of the stable conditions and the presence of pollinators that navigate through this relatively open space. As we continue our vertical journey, we reach the canopy layer, the green ceiling of the rainforest that stretches from about 20 to 40 meters above the ground. This is where the true magic of rainforest architecture becomes apparent. The canopy forms an almost continuous layer of interwoven branches and leaves, creating a complex three-dimensional habitat that supports an astonishing diversity of life. From below, the canopy appears as a solid green roof, but from within, it reveals itself as an intricate world of highways, gardens, and neighborhoods suspended in midair. The canopy captures the vast majority of sunlight that reaches the rainforest, making it the primary energy-gathering zone of the entire ecosystem. Here, massive trees spread their crowns wide, competing for light while also creating the very structure that defines the forest. These canopy trees, known as dominants, include species like mahogany, Brazil nut, and various fig trees, each with their own strategy for claiming their share of the sunday. The physical structure of the canopy creates numerous microenvironments, each with its own unique conditions. The upper surfaces of the canopy are exposed to intense tropical sunlight and can experience temperature fluctuations of 15 degrees Celsius or more throughout the day. In contrast, the inner portions of the canopy remain cooler and more humid, protected by the leaf layers above. This variation creates opportunities for different species to specialize in different parts of the canopy environment. Perhaps nowhere is the nurturing nature of rainforest architecture more evident than in the phenomenon of epiphytism. Epiphytes are plants that grow on other plants without parasitizing them, using their hosts purely for physical support while obtaining their nutrients from the air, rain, and debris that accumulates around them. The complex branching structure of canopy trees provides countless surfaces where epiphytes can establish themselves, from the major forks where large branches divide to the smallest twigs at the canopy's edge. These aerial gardens create entire ecosystems, suspended dozens of meters above the ground. Bromeliads, with their cup-shaped arrangements of leaves, collect rainwater and organic debris, forming tiny ponds that serve as homes for insects, small frogs, and even aquatic plants. Orchids, perhaps the most renowned of epiphytes, have evolved extraordinary adaptations for life in the trees with specialized roots that can absorb moisture directly from humid air and modified stems that store water for dry periods. The architectural complexity of the canopy provides numerous pathways for animals to move through the forest without ever touching the ground. Monkeys swing from branch to branch using their powerful arms and prehensile tails. Birds navigate through the three-dimensional maze with remarkable precision, many species spending their entire lives in the canopy layer. Even terrestrial animals like jaguars and kinkajous are accomplished climbers, using the canopy's highways to move through their territories and hunt for prey. Above the main canopy layer, scattered giant trees pierce through to form the emergent layer, reaching heights of 50 meters or more. These forest giants, the skyscrapers of the rainforest, experience the full intensity of tropical weather. Their crowns are battered by wind and rain, baked by intense sunlight, and must endure the greatest temperature fluctuations in the entire forest. Yet their prominence provides unique advantages, including unlimited access to sunlight and the ability to disperse seeds over vast distances with the help of wind and large birds. Emergent trees often develop distinctive adaptations to cope with their exposed position. Many have buttressed roots, massive triangular supports that extend from the base of the trunk to provide stability in the soft rainforest soil. Their wood tends to be extremely hard and dense, able to withstand the mechanical stress of wind and storms. The crowns of emergent trees are often relatively small in proportion to their height, reducing wind resistance while still providing adequate surface area for photosynthesis. The spacing between these forest giants is crucial to the architecture of the rainforest. Unlike a plantation where trees are planted in neat rows, emergent trees are scattered across the landscape at irregular intervals. This spacing ensures that when an emergent tree eventually falls, it doesn't create a massive gap that would fundamentally alter the forest structure. Instead, the fall of a giant creates a relatively small opening that allows light to penetrate to the lower layers, stimulating growth and regeneration without destroying the forest's overall integrity. The three-dimensional complexity of rainforest architecture creates more than just different levels of habitat. It generates countless microclimates within the forest ecosystem. Temperature gradients exist not only vertically, but horizontally as well, with different parts of the canopy experiencing varying degrees of sun exposure throughout the day. Humidity levels fluctuate based on proximity to water sources, leaf density, and air circulation patterns. Wind patterns are channeled and modified by the forest structure, creating everything from still air pockets to breezy corridors. These microclimatic variations are fundamental to how the rainforest nurtures such extraordinary diversity. A change in elevation of just a few meters can mean the difference between a hot, dry microhabitat and a cool, moist one. Different species of insects might prefer different temperature ranges within the same tree, with some favoring the warmer upper branches, while others thrive in the cooler, more humid conditions of the inner canopy. The light environment created by rainforest architecture is particularly crucial to understanding how this ecosystem nurtures life. As sunlight filters down through the layers of leaves, it becomes increasingly dim and changes in quality as well as quantity. The upper canopy absorbs much of the red and blue light that plants need for photosynthesis, allowing more green light to penetrate to lower levels. This means that understory plants must be particularly efficient at capturing and using the available light, leading to the evolution of specialized photosynthetic strategies and leaf structures. Some plants have evolved leaves with special adaptations for low light conditions, such as larger chloroplasts or modified internal structures that concentrate available light. Others have developed the ability to move their leaves throughout the day to track whatever sunlight penetrates the canopy above. These adaptations demonstrate how the architectural constraints of the rainforest have driven evolutionary innovation, with plants developing increasingly sophisticated strategies for making the most of limited light resources. The vertical structure of the rainforest also influences how water moves through the ecosystem. Rain falling on the emergent layer begins a journey downward that can take hours to complete. Water is intercepted and temporarily stored by epiphytes, channeled along branches and trunks, and gradually released as it makes its way toward the forest floor. This interception and gradual release helps prevent erosion and ensures that water is available to plants throughout the forest, even during brief dry periods. The architecture of the rainforest creates what ecologists call edge effects, where the conditions at the boundary between different layers or habitats differ from those in the interior. These edge environments often support unique communities of plants and animals adapted to the transitional conditions. The edges of canopy gaps, for example, receive more light than the deep understory, but are more protected than the full canopy, creating opportunities for species that require intermediate conditions. As we consider the full scope of rainforest architecture, from the decomposer-rich forest floor to the wind-swept emergent crowns, it becomes clear that this vertical complexity is not simply an accident of growth patterns. It represents millions of years of evolutionary refinement, with each layer optimized to capture and utilize different resources while providing habitat for species with vastly different needs. The forest architecture is both the result of intense competition for resources and a framework that minimizes direct competition by creating numerous distinct niches. This three-dimensional structure serves as the foundation for all other aspects of rainforest ecology. The complex pathways created by branches and vines allow for the movement of animals that pollinate plants and disperse seeds. The varied microclimates support different species assemblages that can coexist in the same geographic area. The multiple layers ensure that resources like light and space are utilized with remarkable efficiency, supporting biomass levels that exceed those of any other terrestrial ecosystem. The architecture of life in the rainforest demonstrates nature's remarkable ability to organize complexity in ways that maximize opportunity and minimize waste. Every layer, every surface, every microhabitat serves multiple functions in supporting the intricate web of relationships that define these ecosystems. As we prepare to explore the next chapter of our rainforest journey, We carry with us an appreciation for how the forest's physical structure creates the stage upon which the drama of tropical ecology unfolds, setting the conditions that allow the rainforest to nurture more forms of life than any other ecosystem on Earth. In the economy of the rainforest, sunlight serves as the fundamental currency, the energy that drives all biological transactions and sustains the incredible abundance of life we've begun to explore. Each photon that penetrates the forest canopy represents a unit of potential energy capable of being transformed through the ancient alchemy of photosynthesis into the chemical bonds that power everything from the smallest bacterium to the mightiest emergent tree. Understanding how this solar economy operates reveals one of nature's most sophisticated systems for capturing, converting, and distributing energy with remarkable efficiency. The tropical sun that bathes the rainforest delivers energy with an intensity and consistency unknown in temperate regions. Near the equator, where most rainforests thrive, the sun's angle remains high throughout the year, providing roughly 12 hours of daylight every single day. This predictable solar schedule has profound implications for how forest plants have evolved their energy gathering strategies. Unlike temperate forests that must cope with dramatic seasonal variations in light availability, tropical rainforest plants can count on a steady supply of solar energy year-round, allowing them to maintain active photosynthesis continuously. The sheer quantity of solar energy striking the rainforest canopy is staggering. On a clear day, each square meter of canopy receives approximately 1,000 watts of solar radiation, enough energy to power several household light bulbs. When multiplied across the vast expanse of tropical forests, this represents one of the largest concentrations of energy capture on Earth. Yet the forest's success lies not just in the abundance of available energy, but in how efficiently this energy is intercepted, converted, and utilized by the myriad plant species that have evolved to exploit every possible niche in the vertical light gradient. At the canopy level, where the forest's energy capture is most intense, trees have evolved remarkable strategies to maximize their photosynthetic efficiency. The leaves of canopy trees are typically smaller and thicker than those of understory plants, adaptations that help them cope with the intense solar radiation and heat of full sun exposure. These leaves often have waxy coatings that reflect excess light and reduce water loss, while their internal structure is optimized for highlight photosynthesis. The arrangement of leaves within canopy tree crowns follows principles that maximize light interception while minimizing self-shading. Leaves are positioned at angles that allow sunlight to penetrate into the crown's interior, ensuring that even leaves deeper within the crown receive enough light for photosynthesis. Some species have evolved compound leaves that can adjust their orientation throughout the day, tracking the sun's movement to maintain optimal light capture while avoiding the damage that can result from excessive radiation. Perhaps the most sophisticated aspect of canopy photosynthesis is how different trees coordinate their crown architecture to minimize direct competition for light. Rather than simply growing taller in an endless race toward the sun, many canopy species have evolved what botanists call crown shyness, a phenomenon where the crowns of adjacent trees rarely touch, creating visible channels of sky between them. This spacing allows each tree to maximize its light capture without completely shading its neighbors, creating a more efficient system for the forest as a whole. The photosynthetic machinery within canopy leaves represents millions of years of evolutionary refinement. Chloroplasts, The cellular organelles where photosynthesis occurs are densely packed in the upper layers of canopy leaves, positioned to capture maximum light while being protected from damage by specialized screening compounds. These leaves often contain multiple types of chlorophyll and accessory pigments that allow them to absorb light across a broader spectrum than plants in low-light environments. Within each chloroplast, the fundamental chemical transformation occurs as light energy drives the conversion of carbon dioxide and water into glucose and oxygen. This process, while familiar in its basic outline, operates with remarkable sophistication in rainforest plants. The enzyme Rubis-Co, which catalyzes the initial step of carbon fixation, is present in enormous quantities in canopy leaves, sometimes comprising up to half of the leaf's total protein content. This massive investment in photosynthetic machinery reflects the intense competition for light resources in the forest canopy. Below the canopy, where light becomes a scarce and precious commodity, plants have evolved entirely different strategies for energy capture. The dappled light environment of the understory, where only 5% of the canopy's illumination penetrates, has driven the evolution of some of nature's most efficient, low-light photosynthetic systems. Understory plants typically have much larger leaves with thinner construction, maximizing their light-gathering surface area while minimizing the energy cost of leaf construction and maintenance. The leaves of understory plants often appear almost black-green, a coloration that results from extremely high chlorophyll concentrations. These plants pack their leaves with photosynthetic machinery to extract energy from every available photon. Their chloroplasts are often larger and more numerous than those of highlight plants, and they may contain specialized arrangements of photosynthetic membranes that enhance light capture efficiency. One of the most remarkable adaptations to low light conditions is the evolution of shade leaves with modified internal architecture. These leaves have enlarged air spaces that scatter light internally, ensuring that photons have multiple opportunities to be absorbed as they bounce around within the leaf. Some understory plants have evolved leaves with specialized cells that act as light guides, channeling whatever light is available directly to the chloroplast-rich photosynthetic tissues. The temporal dynamics of light availability in the understory have led to the evolution of plants that can rapidly adjust their photosynthetic activity to take advantage of brief periods of direct sunlight. When a gap opens in the canopy above, perhaps from a fallen branch, the sudden influx of direct sunlight can increase light levels by 50-fold or more. Understory plants have evolved the ability to rapidly upregulate their photosynthetic machinery to capitalize on these fleeting opportunities, while also possessing protective mechanisms to prevent damage from the sudden increase in light intensity. The climbing plants of the rainforest, the lianas and vines, represent perhaps the most creative solution to the challenge of reaching sunlight in a competitive environment. Rather than investing energy in building a massive trunk to support their own weight, these plants have evolved to use other trees as living scaffolding, devoting their resources to rapid vertical growth rather than structural support. This strategy allows them to reach the canopy using a fraction of the biomass investment required by trees. Lianas exhibit remarkable adaptations for their climbing lifestyle. Many species begin life as seemingly delicate seedlings on the forest floor, but once they locate a suitable support tree, they can grow vertically at astonishing rates. Some tropical lianas can grow more than 20 meters per year, racing toward the light with single-minded determination. Their stems often develop flexible, rope-like properties that allow them to bend and sway with their host trees without breaking. The leaves of climbing plants show fascinating adaptations to their unique lifestyle. Young plants growing in the understory often have large, thin leaves optimized for low-light conditions, but as the same plant reaches the canopy, its leaves may become smaller, thicker, and waxy, adapted for the high-light environment above. This phenotypic plasticity allows a single individual plant to optimize its energy capture across the entire vertical gradient of the forest. Some lianas have evolved even more sophisticated strategies for light capture. Certain species produce different types of leaves at different heights, with shade leaves in the understory and sun leaves in the canopy, sometimes on the same stem. Others develop extensive branching systems once they reach the canopy, spreading their photosynthetic surface across multiple host trees to maximize their light capture area. The epiphytes, those aerial plants that live on the branches and trunks of trees, represent the ultimate expression of the rainforest's ability to exploit every possible opportunity for energy capture. These plants have abandoned the forest floor entirely, establishing themselves directly in the canopy where light is abundant. Yet their elevated lifestyle presents unique challenges, particularly in obtaining water and nutrients that ground-rooted plants can access through their root systems. Epiphytic bromeliads have evolved some of the most ingenious solutions to the challenges of aerial life. Their leaves form cup-shaped rosettes that capture and hold rainwater, creating suspended pools that serve multiple functions. These water reservoirs not only provide moisture for the plant, but also collect organic debris from the forest canopy, which decomposes to provide nutrients. The roots of many epiphytic bromeliads have become specialized for absorbing water and nutrients directly from these leaf-held pools rather than from soil. Orchids, the most diverse family of epiphytes, have evolved extraordinary adaptations for aerial photosynthesis. Many species have developed thick, succulent leaves and stems that store water and nutrients for use during dry periods. Their specialized aerial roots are covered with a spongy tissue called velamen that can rapidly absorb moisture from humid air, allowing these plants to harvest water directly from the atmosphere. Some epiphytic orchids have taken the adaptation to aerial life even further, developing what botanists call CAM photosynthesis, or crassulation acid metabolism. This specialized form of photosynthesis allows plants to open their stomata and collect carbon dioxide during the cool, humid nights, storing it in the form of organic acids. During the hot, dry days, the stomata remain closed to conserve water, while the stored acids are broken down to release carbon dioxide for photosynthesis. This system allows epiphytes to continue photosynthesis even under conditions where water conservation is critical. The diversity of photosynthetic strategies extends to the forest floor, where specialized plants have evolved to function in the permanent twilight of the understory. Some species have developed the ability to photosynthesize using green light, which penetrates the canopy more effectively than red or blue light. Others have evolved partnerships with mycorrhizal fungi that are so extensive that the plant obtains much of its carbon from the fungal partner rather than from its own photosynthesis, essentially becoming partially parasitic on the forest's vast underground fungal network. The efficiency of energy capture in rainforest ecosystems extends beyond individual plant adaptations to encompass system-level optimizations. The multi-layered structure of the forest ensures that light is captured at multiple levels, with each layer specialized for different light conditions. Canopy trees capture the majority of direct sunlight, while understory plants and epiphytes harvest the filtered and reflected light that would otherwise be lost to the system. The temporal coordination of photosynthetic activity across the forest reveals another level of sophistication in the rainforest's energy economy. Different species may have peak photosynthetic activity at different times of day, ensuring that the forest's energy capture systems are active throughout the daylight hours. Some canopy trees may photosynthesize most efficiently during the intense midday sun, while understory plants might maximize their activity during the gentler morning and evening light when the sun's angle allows more penetration through the canopy. The chemical products of photosynthesis flow through the rainforest ecosystem in complex pathways that connect every level of the forest community. The glucose produced by photosynthesis serves not only as food for the plants themselves, but also as the foundation for the elaborate food webs that support the forest's animal communities. The oxygen released as a byproduct of photosynthesis maintains the atmosphere that all forest organisms depend upon for respiration. Perhaps most remarkably, the rainforest's photosynthetic systems have evolved mechanisms for sharing energy resources across species boundaries. The vast networks of mycorrhizal fungi that connect tree roots can transfer carbon compounds between different plant species, creating what some researchers call a wood-wide web of energy sharing. A large tree, photosynthesizing in full sunlight, might share some of its excess carbon with smaller understory plants through these fungal networks, while receiving nutrients that the understory plants are better positioned to acquire. The seasonal rhythms of solar energy, while less dramatic in tropical regions than in temperate zones, still influence the forest's photosynthetic activity. Even near the equator, there are subtle variations in day length and sun angle throughout the year, and many forest plants time their growth and reproductive cycles to coincide with periods of optimal light availability. Some species produce new leaves just before the wettest months, when cloud cover might reduce light levels, maximizing their photosynthetic capacity during challenging periods. As we consider the full scope of the rainforest's solar economy, from the intense photosynthesis of sun-baked canopy leaves to the delicate energy capture of understory herbs, we begin to understand how this ecosystem has achieved such remarkable productivity. The forest has evolved not as a collection of competing individual plants, but as an integrated system for capturing and utilizing solar energy with extraordinary efficiency. The success of this system lies in its diversity of approaches to the fundamental challenge of converting sunlight into usable energy. Every possible niche for photosynthesis has been exploited, from the blazing canopy to the dim forest floor, from the aerial gardens of epiphytes to the climbing pathways of lianas. This diversity ensures that virtually every photon entering the forest has multiple opportunities to be captured and converted into the chemical energy that powers the incredible complexity of rainforest life. The currency of sunlight, transformed through countless variations of photosynthetic chemistry, creates the energy foundation that supports not only the forest's remarkable plant diversity, but also the intricate networks of relationships between plants and the countless other organisms that depend upon them. As we prepare to explore how this captured energy moves through the forest's nutrient cycling systems, we carry with us an appreciation for the sophisticated biological technologies that transform the simple gift of sunlight into the complex energy economy that makes the rainforest Earth's most productive ecosystem. One of the most paradoxical aspects of rainforests lies beneath our feet, hidden in the very foundation that supports this incredible abundance of life. Dig down through the rich layer of decomposing leaves on the forest floor, and you'll discover something surprising. The soils of tropical rainforests are often remarkably poor in nutrients. These ancient soils, weathered by millions of years of intense tropical rains and heat, have been leached of most minerals that plants require for growth. Yet somehow, these nutrient-poor substrates support the most productive terrestrial ecosystems on Earth. The secret lies in one of nature's most sophisticated recycling systems, a biological alchemy that transforms death into life with extraordinary efficiency. The paradox of rainforest productivity begins with understanding the age and nature of tropical soils. Unlike the fertile soils of temperate regions, which are relatively young and rich in minerals from recent glacial activity, tropical soils are often millions of years old. The intense heat and abundant rainfall of tropical climates accelerate chemical weathering processes that break down rock and soil minerals, washing away essential nutrients like nitrogen, phosphorus, and potassium in a process called leaching. What remains after this prolonged weathering is typically a deep layer of clay-rich soil dominated by iron and aluminum oxides, which give many tropical soils their characteristic reddish color. These soils, known as oxasols and ultisols, are often acidic and have limited capacity to hold onto the nutrients that plants need for growth. In many temperate ecosystems, such soil conditions would support only sparse vegetation, yet rainforests have evolved remarkable mechanisms to overcome these constraints. The key to understanding rainforest nutrient cycling lies in recognizing that the vast majority of the ecosystem's nutrients are not stored in the soil, but rather in the living biomass of the forest itself. The trees, understory plants, epiphytes, animals, and microorganisms collectively contain and actively cycle the nutrients that sustain the entire ecosystem. When an organism dies, whether it's a massive canopy tree or a tiny insect, it becomes part of an incredibly efficient recycling system that rapidly breaks down organic matter and makes its nutrients available to living plants. This recycling begins the moment organic matter reaches the forest floor. The warm, humid conditions of the tropical environment create ideal circumstances for decomposition, with temperatures remaining consistently high and moisture levels staying optimal for the activity of decomposer organisms. Bacteria and fungi immediately begin breaking down dead plant and animal tissues, working with remarkable speed compared to their counterparts in cooler climates. The decomposer community of the rainforest represents one of nature's most diverse and efficient processing systems. Countless species of bacteria specialize in breaking down different types of organic compounds, from simple sugars to complex lignins and celluloses. Fungi extend thread-like hyphae through the decomposing matter, secreting enzymes that break down organic molecules and absorb the resulting nutrients. These fungal networks often extend far beyond individual pieces of decomposing matter, connecting living plant roots with decomposing organic material in what ecologists call the soil food web. Perhaps the most remarkable aspect of rainforest decomposition is its speed. A leaf that falls from the canopy may be completely broken down and its nutrients recycled within weeks or months, compared to the years it might take for similar decomposition in temperate forests. This rapid turnover ensures that nutrients don't remain locked up in dead organic matter for long periods, but are quickly made available to support new growth. The efficiency of this decomposition process is enhanced by the incredible diversity of decomposer organisms. Different species of bacteria and fungi specialize in breaking down different types of organic compounds, ensuring that virtually every component of dead organisms can be processed and recycled. Some fungi specialize in breaking down cellulose, the structural compound in plant cell walls, while others focus on lignin, the complex polymer that gives wood its strength. Still others concentrate on proteins, fats, or other organic molecules. Invertebrate decomposers play equally important roles in the nutrient cycling process. Termites, with their powerful jaws and symbiotic gut bacteria, can process vast quantities of dead wood and plant matter. Their colonies can contain millions of individuals, all working together to break down cellulose and other complex organic compounds that many other organisms cannot digest. The constant activity of termite colonies creates a continuous stream of processed organic matter that becomes available to other decomposers and eventually to plants. Millipedes, beetles, and countless other invertebrates contribute to the mechanical breakdown of organic matter, fragmenting large pieces into smaller particles that provide more surface area for bacterial and fungal decomposition. Earthworms and other soil invertebrates mix organic matter with mineral soil, creating intimate contact between decomposing material and the soil particles that can hold nutrients temporarily before plants absorb them. The fungal component of rainforest nutrient cycling deserves special attention for its sophistication and importance. Mycorrhizal fungi form partnerships with plant roots that are fundamental to how nutrients move through the forest ecosystem. These fungi extend the effective reach of plant root systems by orders of magnitude, with their thread-like hyphae exploring soil volumes that roots could never access directly. In exchange for sugars produced by plant photosynthesis, these fungi provide plants with nutrients they've absorbed from the soil and decomposing organic matter. The mycorrhizal networks in rainforests can be truly vast, with individual fungal organisms potentially connecting dozens or even hundreds of trees across large areas of forest. These networks function as nutrient highways, moving resources from areas where they're abundant to areas where they're scarce. A tree growing in a nutrient-rich patch might share excess nutrients with another tree growing in poorer conditions, mediated by the fungal networks that connect them. Recent research has revealed that these fungal networks can even transfer nutrients between different species of plants, challenging our traditional understanding of plant competition. Rather than simply competing for limited resources, plants connected by mycorrhizal networks may actually cooperate, sharing resources in ways that benefit the overall health and stability of the forest ecosystem. Large, established trees might support smaller understory plants through these networks, while younger trees might receive essential nutrients from their larger neighbors. The root systems of rainforest plants have evolved remarkable adaptations for nutrient capture that reflect the challenges of obtaining nutrients from poor soils. Many tropical trees have extensive shallow root systems that spread wide rather than deep, maximizing their ability to capture nutrients from the organic matter that accumulates on the soil surface. These surface roots often form dense mats that can efficiently absorb nutrients as soon as they're released by decomposition processes. Some trees have developed specialized root structures called feeder roots that grow directly into decomposing logs or leaf litter. These roots can extract nutrients directly from organic matter as it decomposes, bypassing the soil entirely. This adaptation is particularly important in very nutrient-poor soils, where traditional root uptake from soil solutions might be insufficient to support large trees. Buttress roots, the massive triangular supports that extend from the bases of many large tropical trees, serve multiple functions in nutrient cycling. While their primary role is structural support, these roots also create catchment areas where organic matter accumulates, concentrating nutrients near the tree where they can be readily absorbed. The crevices between buttress roots often become gardens of decomposing matter that feed the tree's extensive surface root system. The timing of nutrient release and uptake in rainforests shows remarkable coordination between different components of the ecosystem. Many trees time their leaf production and root growth to coincide with periods when nutrients are most readily available. Some species produce new leaves just before the peak of the wet season, when rapid decomposition will make nutrients most abundant. Others coordinate their root growth with the fall of nutrient-rich litter from canopy trees. Epiphytes have evolved particularly creative solutions to the challenges of obtaining nutrients without access to soil. Many epiphytic bromeliads have modified their leaves to form tank-like structures that collect not only rainwater, but also organic debris falling from the canopy above. These aerial gardens support entire communities of bacteria and small invertebrates that break down the collected organic matter, making nutrients available to the bromeliad while creating habitat for other organisms. The roots of epiphytic orchids and other air plants have developed the ability to absorb nutrients directly from dust, pollen, and organic particles suspended in the atmosphere. These specialized roots are covered with a spongy tissue that can capture and concentrate dilute nutrients from sources that terrestrial plants cannot access. Some epiphytes have even evolved partnerships with ants, providing housing for ant colonies in exchange for the nitrogen-rich waste products the ants produce. The nutrient cycling systems of rainforests operate at multiple temporal scales, from the rapid cycling that occurs over days and weeks to longer-term cycles that span years or decades. The rapid cycles involve the breakdown and uptake of easily decomposed organic matter like fallen leaves and fruit. Medium-term cycles process more resistant materials like branches and bark, while long-term cycles handle the most persistent organic compounds and the gradual weathering of mineral components. Water plays a crucial role in rainforest nutrient cycling, serving as both a transport medium and a driver of chemical processes. The abundant rainfall in tropical regions leaches nutrients from surfaces and transports them through the soil, but the forest has evolved mechanisms to minimize nutrient loss. The dense canopy intercepts much of the rainfall, allowing it to trickle slowly through the forest layers and giving plants maximum opportunity to absorb nutrients before they're carried away. Stem flow, the water that runs down tree trunks during rain events, becomes concentrated with nutrients leached from bark, epiphytes, and other organic matter on the tree surfaces. This nutrient-enriched water is delivered directly to the base of trees, where their root systems can efficiently absorb it. Some trees have evolved bark patterns and canopy structures that maximize the capture and concentration of stem flow nutrients. The chemical complexity of rainforest nutrient cycling involves the transformation of nutrients between different chemical forms as they move through the ecosystem. Nitrogen, one of the most important limiting nutrients in many ecosystems, cycles through numerous chemical forms in tropical forests. It may exist as organic nitrogen in proteins and nucleic acids, as ammonium ions in decomposing matter, as nitrate ions in soil solutions, or as nitrogen gas in the atmosphere. Specialized bacteria and fungi mediate many of these nitrogen transformations. Nitrogen-fixing bacteria convert atmospheric nitrogen gas into forms that plants can use, while other bacteria convert organic nitrogen from decomposing matter into mineral forms. Still, other microorganisms complete the nitrogen cycle by converting excess nitrates back to nitrogen gas, preventing the accumulation of potentially toxic nitrogen compounds. Phosphorus cycling in rainforests presents particular challenges because this essential nutrient is often extremely limited in tropical soils. Plants have evolved highly efficient mechanisms for capturing and recycling phosphorus, including specialized root structures that can extract phosphorus from organic compounds and close partnerships with fungi that excel at mobilizing phosphorus from mineral sources. The efficiency of rainforest nutrient cycling extends beyond individual nutrients to encompass complex interactions between different elements. The cycling of carbon through photosynthesis and decomposition drives many other nutrient cycles, while the availability of nutrients like nitrogen and phosphorus can limit carbon cycling. These interconnected cycles create feedback loops that help maintain the stability and productivity of the forest ecosystem. Seasonal patterns in rainfall and temperature create rhythms in nutrient cycling that many forest organisms have synchronized their life cycles to match. Periods of intense rainfall may flush nutrients through the system, while drier periods allow for the accumulation of organic matter. Many plants time their reproductive cycles to coincide with periods of optimal nutrient availability, ensuring that developing seeds and fruits have access to the resources they need. The remarkable efficiency of rainforest nutrient cycling represents millions of years of evolutionary refinement, with each component of the system optimized to capture, process, and redistribute the limited nutrients available in these ecosystems. The result is a biological recycling system that operates with virtually no waste, transforming the death of every organism into the nutrients that sustain new life. This efficient recycling creates the foundation that supports the incredible diversity and productivity of rainforest ecosystems, demonstrating how biological systems can overcome environmental limitations through cooperation, specialization, and remarkable efficiency in resource use. Water is the lifeblood of the rainforest, flowing through every aspect of this remarkable ecosystem in ways both obvious and subtle. From the thunderous downpours that can unleash 30 centimeters of rain in a single day to the microscopic droplets that condense on spider webs at dawn, water shapes every interaction, every adaptation, and every relationship within the forest. Unlike other ecosystems that must carefully conserve this precious resource, rainforests exist in a world of aquatic abundance, where the challenge is not finding water, but managing its overwhelming presence. The water cycle in tropical rainforests operates on a scale that dwarfs similar processes in other ecosystems. Annual rainfall in many tropical forests exceeds 2,000 millimeters, with some regions receiving more than 4,000 millimeters per year. To put this in perspective, the wettest rainforests receive more rain in a single month than many deserts see in a decade. This enormous input of water from the atmosphere creates the foundation for all other ecological processes in the forest, driving everything from nutrient cycling to plant growth to animal behavior. The arrival of water in rainforests is often dramatic and intense. Tropical storms can develop rapidly, transforming a sunny morning into a deluge within minutes. The sound of approaching rain in a rainforest is unmistakable, beginning as a distant roar as millions of raindrops strike leaves in the canopy, gradually building to a crescendo that can make conversation impossible. These intense precipitation events can deliver water at rates that exceed the soil's capacity to absorb it, creating temporary streams and waterfalls where none existed moments before. Yet the forest has evolved remarkable mechanisms for managing these extreme water inputs. The multi-layered canopy acts as a natural umbrella system, intercepting rainfall and moderating its impact on the forest floor. Large leaves in the canopy can catch and temporarily hold substantial amounts of water, releasing it gradually as small streams that trickle down branches and trunks. This process, known as throughfall and stem flow, transforms violent downpours into gentler, more manageable water delivery that the forest ecosystem can better utilize. The interception of rainfall by the forest canopy creates unique microenvironments throughout the vertical structure of the forest. Epiphytes living in the canopy have evolved to take advantage of this intercepted water, with many species developing specialized structures to capture and store the water that flows past their perches. Tank bromeliads create miniature aquatic ecosystems dozens of meters above the ground, their overlapping leaves forming reservoirs that can hold several liters of water and support entire communities of aquatic insects, small frogs, and microscopic organisms. The journey of water through the rainforest begins long before it reaches the ground. As precipitation moves through the canopy layers, it picks up nutrients leached from leaves, bark, and the countless epiphytes that populate the trees. By the time water reaches the forest floor, it has been enriched with dissolved organic compounds, minerals, and other substances that make it a dilute nutrient solution rather than pure water. This enrichment process ensures that every drop of rain carries resources that can be immediately utilized by the forest's living systems. The forest floor itself represents a highly specialized environment for water management. The thick layer of decomposing organic matter that carpets the ground acts like an enormous sponge, capable of absorbing and holding tremendous quantities of water. This organic layer can absorb water at rates many times faster than the underlying mineral soil, preventing erosion and providing a reservoir that slowly releases moisture during drier periods. The complex three-dimensional structure of decomposing leaves, twigs, and other organic matter creates countless tiny spaces where water can be temporarily stored and gradually released. Beneath this organic layer, the root systems of rainforest plants have evolved remarkable adaptations for water uptake that reflect both the abundance and the unpredictability of water availability. Many large trees develop extensive shallow root systems that spread far beyond their canopy edges, creating wide nets for capturing water as it moves across the forest floor. These surface roots often form dense mats that can quickly absorb water from even light rains before it has a chance to run off or evaporate. The relationship between rainforest plants and water extends far beyond simple uptake and utilization. Through the process of transpiration, plants actively participate in the forest's water cycle, releasing water vapor from their leaves back into the atmosphere, where it can contribute to the formation of new clouds and precipitation. A single large canopy tree can transpire hundreds of liters of water per day, creating its own local humidity cloud that influences temperature and moisture conditions in its immediate vicinity. The collective transpiration of the entire forest creates what meteorologists call the evapotranspiration feedback loop, where the forest essentially generates its own rainfall. Water absorbed by plant roots is transported up through stems and released from leaves into the atmosphere, where it rises with warm air currents and eventually condenses to form clouds. These locally formed clouds often produce rainfall that falls back on the forest, creating a self-sustaining precipitation system that can operate independently of weather patterns originating far from the forest. This self-generated rainfall is particularly important during seasonal dry periods when precipitation from external weather systems may be reduced. The forest's ability to create its own local weather patterns helps maintain the high humidity and frequent precipitation that its plant and animal communities depend upon. Research has shown that large tropical forests can generate up to half of their own rainfall through evapotranspiration, making them remarkably self-sufficient in terms of water supply. The movement of water through rainforest landscapes creates a complex network of aquatic habitats that support specialized communities of plants and animals. Temporary pools formed during heavy rains become breeding sites for countless species of frogs, insects, and other organisms that have evolved life cycles synchronized with these ephemeral water bodies. Some species of frogs can complete their entire development from egg to adult in temporary pools that exist for only a few weeks, while others have evolved extended developmental periods that allow them to survive if their pools persist longer than usual. Permanent streams and rivers that flow through rainforests represent major corridors for both water and nutrients moving through the ecosystem. These waterways carry dissolved minerals, weathered from rocks upstream, organic matter washed from the surrounding forest, and sediments that will eventually be deposited downstream. The chemistry of rainforest streams is typically quite different from that of streams in other ecosystems, reflecting the influence of the surrounding forest's biological processes. The interaction between forest streams and the surrounding terrestrial ecosystem creates some of the most dynamic interfaces in the rainforest. Streamside vegetation often differs markedly from that of the surrounding forest, adapted to the higher light levels and different moisture conditions found along waterways. These riparian zones serve as corridors for animal movement and often support different plant communities than the surrounding forest interior. Many rainforest streams are characterized by their dark, tea-colored water, which results from dissolved organic compounds leached from the surrounding forest. These tannins and other organic substances create acidic conditions that influence which organisms can survive in the water and affect the stream's chemistry and ecology. Some fish species have evolved specific adaptations for life in these acidic waters, while others require the more neutral conditions found in streams with different chemical characteristics. The seasonal patterns of rainfall in many tropical regions create dramatic changes in stream flow and water levels that forest organisms have learned to exploit. During wet seasons, small streams can become raging torrents, while dry seasons may reduce them to isolated pools or cause them to disappear entirely. Fish populations may concentrate in the remaining deep pools during dry periods, creating feeding opportunities for terrestrial predators that normally wouldn't encounter aquatic prey. Waterfalls and rapids created by the steep topography of many rainforest regions generate unique microenvironments characterized by constant moisture from spray and mist. These areas often support specialized plant communities that include species found nowhere else in the forest. The constant moisture and high humidity around waterfalls create conditions that some plants require for reproduction, with certain orchids and other epiphytes flowering only in the immediate vicinity of falling water. The root systems of plants growing near streams and rivers have evolved sophisticated strategies for dealing with periodic flooding and variable water levels. Some trees develop pneumatophores, specialized root structures that can absorb oxygen from the air when normal root systems are submerged. Others have flexible root systems that can bend with floodwaters without breaking, or extensive shallow root networks that can quickly absorb nutrients deposited by floodwaters. Animals in rainforest ecosystems have evolved remarkable adaptations for life in this water-rich environment. Many mammals, birds, and reptiles are excellent swimmers, capable of crossing rivers and navigating flooded areas with ease. Some species, like river otters and certain primates, have become so adapted to aquatic life that they spend much of their time in or near water bodies, feeding on fish and other aquatic organisms. The abundance of water in rainforests creates opportunities for animals to exploit aquatic resources, even if they primarily live in terrestrial habitats. Many birds that normally feed on insects or fruit will wade in shallow streams to catch fish or aquatic invertebrates, while some mammals have learned to dig in stream banks to access aquatic insect larvae and other buried organisms. These flexible feeding strategies reflect the diversity of resources available in water-rich environments. Insects in rainforest environments show extraordinary diversity in their relationships with water. Aquatic larvae develop in streams, temporary pools, and even the small water reservoirs held by epiphytic plants high in the canopy. Some species have evolved complex life cycles that take advantage of different aquatic habitats at different stages of development, while others remain aquatic throughout their lives in the diverse water bodies scattered throughout the forest. The microclimatic effects of abundant water extend throughout the rainforest ecosystem, influencing everything from seed germination to animal behavior. High humidity levels prevent many plants from drying out and allow some species to absorb water directly from the air through specialized leaf structures. The constant presence of moisture also influences decomposition rates, with organic matter breaking down more rapidly in humid conditions than in drier environments. Water availability affects the timing of many biological processes in rainforest ecosystems. Many trees time their leaf production to coincide with the beginning of wet seasons, when abundant water will support rapid growth. Similarly, many animal species coordinate their reproductive cycles with seasonal rainfall patterns, ensuring that offspring are born when water and food resources are most abundant. The storage of water within living tissues is another important adaptation to the feast or famine nature of water availability in some rainforest regions. Succulent epiphytes store water in thick, fleshy leaves or stems that can sustain them through brief, dry periods. Some trees develop water storage tissues in their trunks or branches, creating internal reservoirs that buffer against temporary water shortages. The chemical composition of rainwater in tropical regions can vary dramatically depending on atmospheric conditions and the presence of dust, pollen, or other particles in the air. During certain seasons, rainwater may be enriched with minerals blown in from distant sources, providing pulses of nutrients that many forest organisms have evolved to exploit. Some epiphytes appear to time their growth spurts to coincide with these nutrient-rich rainfall events. The interaction between water and the forest's three-dimensional structure creates complex patterns of moisture distribution that influence where different species can survive. Microclimatic gradients in humidity and moisture availability occur over distances of just a few meters, allowing for the coexistence of species with very different water requirements within the same general area of forest. As water moves through the rainforest ecosystem, from its arrival as precipitation through its journey along streams and rivers toward the ocean, it connects all components of the forest in an intricate web of hydrological relationships. Every organism depends on this flow of water, and most participate in it either by intercepting, storing, transpiring, or otherwise influencing the movement of water through the ecosystem. This pervasive influence of water creates the liquid foundation upon which all other rainforest processes depend, setting the stage for the complex, symbiotic relationships that we will explore in our next chapter. In the intricate tapestry of rainforest life, individual survival often depends not on fierce competition, but on sophisticated partnerships that blur the boundaries between separate organisms. These symbiotic relationships, where different species form intimate associations for mutual benefit, represent some of evolution's most elegant solutions to the challenges of life in complex ecosystems. From the microscopic fungal threads that connect tree roots across vast distances to the colorful birds that maintain cleaning stations for other animals, the rainforest is built upon a foundation of cooperation that demonstrates how interconnection creates strength and resilience. The concept of symbiosis encompasses relationships where organisms of different species live in close physical proximity, often with one partner living on or inside the other. While some symbiotic relationships involve one partner benefiting at the expense of another, the most remarkable examples in rainforests are mutualistic partnerships, where both participants gain significant advantages from their association. These relationships often become so intimate and essential that the partners can no longer survive independently, having evolved into biological units that function as integrated wholes. Perhaps nowhere is this interconnectedness more fundamental than in the partnerships between plants and fungi that operate largely unseen beneath the forest floor. Mycorrhizal associations, relationships between plant roots and specialized fungi, form the hidden internet of the rainforest, connecting individual trees and plants in networks that span entire forest ecosystems. These fungal partners extend their thread-like hyphae through the soil, effectively multiplying the surface area of a plant's root system by hundreds or even thousands of times. The mechanics of mycorrhizal partnerships reveal the elegant reciprocity that characterizes the best symbiotic relationships. The fungi, lacking the ability to photosynthesize, receive sugars and other organic compounds from their plant partners, who produce these energy-rich molecules through photosynthesis. In return, the fungi provide their plant partners with minerals and nutrients extracted from the soil, particularly phosphorus and nitrogen, which are often limiting factors for plant growth in tropical ecosystems. But mycorrhizal relationships extend far beyond simple nutrient exchange. The fungal networks that connect multiple plants can transfer resources between different individuals and even between different species, creating a system of mutual support that helps maintain forest stability. When a young seedling struggles to establish itself in the dim understory, it may receive crucial support through mycorrhizal connections to larger, more established trees. Conversely, when old trees are stressed by disease or environmental challenges, they may be supported by nutrients shared through the fungal network from healthier neighbors. Recent research has revealed that these fungal networks can even facilitate communication between plants, transmitting chemical signals that warn of insect attacks, drought stress, or other threats. When one tree is attacked by herbivorous insects, it may release chemical compounds into the mycorrhizal network that trigger defensive responses in neighboring trees, allowing them to begin producing protective compounds before the threat reaches them. This early warning system demonstrates how symbiotic partnerships can enhance survival not just for individual participants, but for entire forest communities. The diversity of mycorrhizal associations in rainforests reflects the evolutionary refinement of these partnerships over millions of years. Different types of fungi form specialized relationships with different groups of plants, creating a complex web of underground associations. Some fungi form structures called arbuscules that penetrate plant root cells, creating intimate interfaces for nutrient exchange. Others form mantles around root tips, enveloping them in fungal tissue that serves as the primary site of interaction between the partners. Above ground, the partnerships between plants and their pollinators create some of the most visually spectacular examples of mutualistic relationships in rainforest ecosystems. The extraordinary diversity of flowering plants in tropical forests has co-evolved with an equally remarkable diversity of animal pollinators, creating intricate partnerships that often involve extreme specialization between particular plant and animal species. Orchids provide some of the most striking examples of specialized pollination partnerships. Many orchid species have evolved flowers that can only be pollinated by a single species of bee, wasp, or other insect. The flowers produce specific chemical attractants that draw their designated pollinators, while the shape and structure of the flower ensures that only the correct pollinator can access the nectar rewards and simultaneously transfer pollen. Some orchids have evolved flowers that mimic female insects so precisely that male insects attempt to mate with them, inadvertently picking up pollen in the process. The partnership between fig trees and their specialized wasp pollinators represents one of the most intricate coevolutionary relationships known to science. Each species of fig tree is pollinated exclusively by a single species of tiny wasp, and each wasp species can only complete its life cycle within the fruits of its specific fig partner. Female wasps enter the closed fig through a tiny opening, losing their wings in the process and becoming trapped inside. They lay their eggs within the fig, while simultaneously pollinating the fig flowers with pollen they carry from other fig trees. The fig provides a nursery for the developing wasps, with some of the fig's flowers developing into seeds, while others become food for the wasp larvae. When the new generation of wasps emerges, the males mate with the females and then die within the fig, while the fertilized females collect pollen and search for exit holes that the males have chewed through the fig wall. The females then fly away to find other fig trees where they can continue the cycle, carrying with them the pollen that will fertilize their next host. This partnership between figs and wasps has persisted for over 60 million years, with both partners evolving in lockstep to maintain their intricate interdependence. The relationship is so precise that the timing of fig fruit development is synchronized with wasp life cycles, and the chemical signals that attract wasps are species-specific, ensuring that each wasp finds its correct partner fig species, even in forests containing dozens of different fig species. Pollination partnerships extend beyond insects to include birds, bats, and other vertebrates that have evolved to serve as pollinators for different plant species. Hummingbirds form partnerships with flowers that produce nectar in tube-shaped corollas, perfectly matched to the length and curve of the bird's bill. The flowers often have bright red or orange colors that are particularly attractive to birds, while their lack of scent reflects the fact that birds rely primarily on vision rather than smell to locate food sources. Bat-pollinated flowers typically open at night and produce strong, musky scents that can attract bats from considerable distances. These flowers are often large and robust, able to withstand the vigorous feeding behavior of bats, and they typically produce copious amounts of nectar to reward their flying mammal pollinators. Many bat-pollinated plants position their flowers away from leaves and branches to provide clear flight paths for their nocturnal partners. The partnerships involved in seed dispersal create another layer of interconnection that links rainforest plants with their animal partners in relationships crucial for forest regeneration and expansion. Many rainforest plants produce fleshy fruits specifically designed to attract animal dispersers, packaging their seeds in nutritious rewards that encourage animals to transport them away from the parent plant. This dispersal is essential for plant reproduction as it reduces competition between parent plants and their offspring and allows plants to colonize new areas of suitable habitat. The great variety of fruit types found in rainforests reflects the diversity of animal dispersers and their different capabilities and preferences. Large fruits with hard shells may be designed for dispersal by large mammals that can crack them open and carry the seeds over long distances. Smaller, colorful fruits attract birds that can carry seeds to new locations during their daily movements through the forest. Some plants produce fruits that ripen at different times to attract different types of dispersers, maximizing their chances of successful seed transport. Primates serve as particularly important seed dispersers for many rainforest plants, often swallowing fruits whole and depositing intact seeds in their feces as they move through the forest. The digestive process may actually improve germination success for some seeds by softening their outer coats or providing nutrients for initial growth. Some plants have evolved fruits that ripen specifically when certain primate species are most active in their areas, timing their reproductive efforts to coincide with the presence of their most effective dispersal partners. Birds create partnerships with plants that reflect their flight capabilities and feeding behaviors. Species that can fly long distances may disperse seeds across large areas, while species with smaller home ranges provide more localized dispersal services. Some bird species have digestive systems that process fruits quickly, depositing seeds relatively close to where they were consumed, while others retain seeds longer, carrying them greater distances before deposition. The cleaning stations maintained by certain bird species represent a different type of mutualistic partnership that demonstrates how service relationships can evolve between very different types of animals. Oxpeckers and similar bird species form partnerships with large mammals, feeding on ticks, flies, and other parasites that infest their partner's skin. The birds receive nutrition from these parasites, while the mammals benefit from the removal of harmful organisms that can cause disease or discomfort. These cleaning relationships often involve sophisticated communication between partners. The mammals may adopt specific postures or positions that allow the cleaning birds better access to areas where parasites are concentrated and last proved. Some mammals appear to actively seek out cleaning stations, traveling considerable distances to locations where they know cleaning birds can be found. The birds, in turn, may follow herds of mammals, maintaining long-term relationships with individual animals whose parasitic loads they regularly manage. Ant-plant partnerships represent some of the most complex, mutualistic relationships in rainforest ecosystems, involving plants that provide housing and food for ant colonies in exchange for protection from herbivores and sometimes nutrients from ant waste. Cecropia trees form partnerships with Azteca ants, providing hollow stems where the ants can build their nests and specialized food bodies that serve as protein sources for the ant colonies. In return, the ants aggressively defend their host trees from insects, mammals, and other animals that might damage the plants. The partnership between acacia trees and acacia ants involves even more intimate codependence. The trees produce hollow thorns that serve as nesting sites for the ants, along with protein-rich food bodies and energy-rich nectar that meet all the nutritional needs of their ant partners. The ants, in return, attack any organism that touches their host tree, clearing away competing vegetation that might shade or crowd the acacia and patrolling constantly for signs of threat. Some ant-plant partnerships involve plants that have evolved specialized structures specifically to house and feed their ant partners. Myrmecodia plants develop swollen stems with internal chambers that provide nesting space for ants while producing food bodies and nectar rewards that sustain their protectors. The ants not only defend these plants from herbivores, but also contribute nutrients through their waste products, which the plants can absorb directly through specialized tissues lining the ant chambers. The partnerships between different animal species create additional layers of interconnection within rainforest communities. Mixed species foraging flocks, where different bird species travel together through the forest, provide mutual benefits through shared vigilance for predators and increased efficiency in locating food sources. Some species in these flocks specialize in detecting and warning others about approaching threats, while others excel at finding food sources that can be shared or that create foraging opportunities for their partners. Cleaner fish relationships in rainforest streams and rivers mirror the cleaning bird partnerships found among terrestrial animals. Small fish species establish cleaning stations where larger fish come to have parasites and dead tissue removed. These relationships involve complex behavioral adaptations, with the clients adopting specific postures to indicate their desire for cleaning services, while the cleaner fish use specialized mouth structures to remove parasites without harming their clients. The temporal coordination of symbiotic relationships adds another dimension to these partnerships, with many species synchronizing their life cycles to maximize the benefits they provide to each other. Plants may time their flowering or fruit production to coincide with the breeding cycles of their pollinators or dispersers, ensuring that their partners have access to resources when they need them most. Similarly, some cleaning relationships intensify during breeding seasons when the health benefits of parasite removal are particularly crucial for reproductive success. The stability and resilience these partnerships provide to rainforest ecosystems become apparent when we consider how the loss of one partner can cascade through entire ecological networks. When a pollinator species declines or disappears, its plant partners may lose their ability to reproduce, eventually disappearing from the ecosystem. This can then affect the animals that depend on those plants for food or shelter, creating ripples of impact that spread far beyond the original partnership. The redundancy built into many symbiotic networks helps buffer against such losses, with most plants having relationships with multiple pollinators or dispersers, and most animals maintaining partnerships with multiple plant species. This redundancy ensures that the loss of any single partnership, while significant, is unlikely to cause the complete collapse of the broader ecological network. These intricate webs of partnership and mutual dependence reveal how rainforest ecosystems achieve their remarkable stability and productivity through cooperation rather than competition alone. The countless symbiotic relationships that connect species across the forest create a resilient network that can adapt to environmental changes while maintaining the essential functions that sustain the entire ecosystem, preparing us to explore how these partnerships contribute to the forest's role as nature's great recycling center. Beneath the spectacular displays of flowering plants and the dramatic interactions of large animals lies the rainforest's most essential workforce, a vast army of organisms dedicated to the unglamorous but crucial task of breaking down the dead and transforming it back into the building blocks of life. This decomposer community operates with remarkable efficiency in the warm, humid conditions of tropical forests, processing organic matter at rates that dwarf similar processes in temperate ecosystems. Without these tireless recyclers, the rainforest's remarkable productivity would be impossible, as nutrients would remain locked away in dead tissue rather than being made available to support new growth. The decomposer's domain encompasses everything from microscopic bacteria and fungi to highly organized societies of insects that have evolved sophisticated systems for processing organic matter. These organisms have colonized every possible niche where decomposition can occur, from the surface of fallen leaves to the interior of rotting logs, from the rich organic layer of the forest floor to the specialized chambers within insect colonies. Together, they form processing networks of extraordinary complexity that ensure virtually no organic material goes to waste in the rainforest ecosystem. The physical conditions of tropical rainforests create an ideal environment for decomposition processes. The consistently warm temperatures, typically ranging between 24 and 28 degrees Celsius, accelerate the chemical reactions involved in breaking down organic compounds. The high humidity levels, often approaching saturation, prevent organic matter from drying out and becoming temporarily resistant to decomposition. The combination of warmth and moisture creates conditions where decomposer organisms can remain active year-round, processing organic matter continuously rather than being limited by seasonal constraints that affect decomposition in temperate regions. Bacterial decomposers form the foundation of the rainforest's recycling system, breaking down organic matter at the molecular level through the secretion of specialized enzymes. Different bacterial species have evolved to process different types of organic compounds, creating an assembly line effect where complex materials are broken down step by step into simpler substances that can be absorbed by plants or processed further by other decomposers. Some bacteria specialize in breaking down cellulose, the structural component of plant cell walls, while others focus on proteins, fats, or the complex lignin compounds that give wood its strength. The diversity of bacterial decomposers in rainforest soils is staggering, with a single gram of forest floor soil containing millions of individual bacteria representing thousands of different species. This diversity ensures that virtually any organic compound that enters the system can be processed by organisms specifically adapted to break it down. The rapid turnover of bacterial populations, with generation times measured in hours rather than days or years, allows these communities to quickly adapt to changing conditions and available resources. Fungal decomposers complement the work of bacteria by specializing in breaking down some of the more resistant organic compounds found in plant tissues. Fungi excel at decomposing lignin and cellulose, the structural compounds that give plants their strength but are difficult for many other organisms to digest. The thread-like hyphae of fungi can penetrate deep into organic matter, secreting enzymes that break down complex molecules and absorbing the resulting nutrients. The fungal networks that spread through decomposing organic matter create three-dimensional webs that connect different pieces of dead material across the forest floor. These networks can rapidly redistribute nutrients from areas where decomposition is producing excess resources to areas where nutrients are scarce, optimizing the efficiency of the entire decomposition system. Some fungi form specialized structures called rhizomorphs, root-like bundles of hyphae that can transport nutrients over distances of several meters, effectively creating a circulatory system within the decomposing organic layer. Wood decomposing fungi demonstrate remarkable specialization in their approaches to breaking down different types of plant material. Brown rot fungi focus on breaking down cellulose and hemicellulose while leaving lignin relatively intact, creating the characteristic brown, crumbly texture of heavily decayed wood. White rot fungi take a different approach, breaking down lignin along with other wood components, often leaving behind white fibrous remnants. Still other fungi specialize in the soft rot decay of wood that occurs in very wet conditions, creating small cavities within wood cells rather than breaking down entire tissues at once. The invertebrate decomposer community includes some of the most sophisticated processing systems found in any ecosystem. Termites represent perhaps the most remarkable example of social organization applied to decomposition, with colonies that can contain millions of individuals working together to process vast quantities of dead plant material. Different termite species have evolved to exploit different types of organic matter, from fresh leaf litter to highly decayed wood, creating a succession of processing stages that maximizes the extraction of nutrients from dead plant tissues. The internal structure of termite colonies reveals the complexity of their decomposition operations. Worker termites collect organic matter from throughout their territory and bring it back to the colony where it is processed through multiple stages. Some workers chew the material into smaller fragments, while others mix it with salivary secretions that begin the breakdown process. Still others tend specialized chambers where the processed organic matter is further decomposed by symbiotic bacteria and fungi that live within the colony structure. The relationship between termites and their microbial partners represents one of the most sophisticated examples of cooperative decomposition. Termite workers cannot directly digest cellulose, the main component of the plant material they collect, but their guts contain specialized chambers housing billions of bacteria and protozoans that can break down cellulose into simpler compounds that the termites can absorb. This partnership allows termites to exploit a food source that would otherwise be unavailable to them while providing their microbial partners with a stable environment and constant supply of raw materials. Different termite species have evolved partnerships with different microbial communities, allowing them to specialize in processing different types of plant material. Some species focus on fresh leaf litter with high nitrogen content, while others specialize in processing woody material that is low in nutrients but abundant in the forest environment. Still others have evolved to process soil organic matter, creating extensive tunnel systems that help mix and aerate forest soils while extracting nutrients from the organic components. Ant societies have also evolved sophisticated approaches to decomposition though their strategies differ significantly from those of termites. Leafcutter ants create some of the most complex agricultural systems found in nature, harvesting fresh leaves not to eat directly, but to use as substrate for growing fungal gardens within their colonies. These ants cut leaves into small fragments and carry them back to their nests, where specialized workers process them further, chewing them into pulp and mixing them with their own secretions to create optimal growing conditions for their fungal partners. The fungal gardens maintained by leafcutter ants represent intensive decomposition operations that convert fresh plant material into easily digestible fungal tissue. The ants carefully tend these gardens, removing contaminating organisms that might interfere with their chosen fungal species and maintaining optimal temperature and humidity conditions for fungal growth. The fungi, in turn, break down the complex plant compounds in the leaf material and convert them into nutritious tissue that serves as the primary food source for the ant colony. The waste products from these fungal gardens are not discarded, but are processed through additional stages of decomposition. Specialized workers remove exhausted substrate from the gardens and deposit it in compost chambers, where different microbial communities continue to break down the remaining organic matter. Eventually, even these final waste products are moved to disposal chambers, where they undergo final decomposition stages before their nutrients are completely recycled back into the forest ecosystem. Beetle larvae form another crucial component of the decomposer community, specializing in breaking down different types of organic matter throughout their development. Many species spend their entire larval stage within decaying logs or other organic matter, creating extensive tunnel systems as they consume the substrate around them. These tunnels not only process the organic matter directly, but also create channels that allow air and water to penetrate deeper into decomposing materials, accelerating the work of bacteria and fungi. The succession of beetle species that colonize decomposing logs reveals the complexity of invertebrate decomposition processes. Early colonizers may specialize in processing fresh organic matter with high moisture content, while later arrivals focus on drier, more decomposed material. Some species create galleries just beneath the bark of dead trees, while others tunnel deep into the heartwood. This succession ensures that every component of dead trees is eventually processed and recycled. Millipedes contribute to decomposition through their role as primary processors of leaf litter, breaking large pieces of organic matter into smaller fragments that provide more surface area for bacterial and fungal attack. Their digestive systems are specifically adapted for processing plant material, with multiple stomach chambers that house different microbial communities specialized for breaking down different components of plant tissue. The fragments of partially processed organic matter that millipedes deposit as waste provide ideal substrates for further decomposition by other organisms. The timing and coordination of decomposer activities create complex temporal patterns that maximize the efficiency of nutrient recycling. Many decomposer organisms have evolved life cycles that synchronize with seasonal patterns in the availability of organic matter. Certain fungal species may become more active during periods when particular types of plant material are most abundant, while some invertebrate decomposers time their reproductive cycles to coincide with peaks in resource availability. The chemical complexity of decomposition processes involves the sequential breakdown of organic compounds, from complex molecules to simple ions that plants can absorb. Proteins are broken down into amino acids and then into ammonia or nitrate ions that serve as nitrogen sources for plants. Complex carbohydrates are converted into simple sugars and eventually into carbon dioxide and mineral nutrients. Lignin, one of the most resistant organic compounds, is slowly broken down into simpler aromatic compounds that may persist in soil for extended periods or be further processed by specialized organisms. The spatial organization of decomposition creates distinct zones within the forest ecosystem where different types of processing occur. The surface layer of fresh organic matter supports different decomposer communities than the partially decomposed material beneath it, which in turn differs from the highly processed organic matter near the mineral-soil interface. This vertical stratification maximizes the efficiency of decomposition by ensuring that each stage of the process occurs under optimal conditions. The interaction between decomposition and the physical structure of the forest creates feedback loops that influence both processes. Decomposition produces the nutrients that support plant growth, while the plants produce the organic matter that feeds decomposer communities. The three-dimensional structure of the forest influences decomposition by affecting temperature, humidity, and airflow patterns that determine where different types of decomposition can occur most efficiently. Seasonal variations in decomposition rates reflect changes in temperature, moisture, and the availability of fresh organic matter. During periods of high rainfall, decomposition may accelerate due to increased moisture availability, while drier periods may slow some decomposition processes while favoring others that are adapted to lower moisture conditions. The timing of leaf fall, fruit drop, and other inputs of organic matter creates pulses of resources that decomposer communities must process efficiently to prevent the accumulation of organic waste. The products of decomposition extend beyond simple nutrients to include complex organic compounds that influence soil chemistry and plant growth in subtle but important ways. Humic acids produced during decomposition can affect soil pH and nutrient availability, while various organic compounds may serve as growth regulators or signaling molecules that influence plant development. Some decomposition products may inhibit the growth of certain plant species while promoting others, contributing to the complex patterns of species composition found in different areas of the forest. The efficiency of rainforest decomposition systems becomes apparent when compared to similar processes in other ecosystems. A leaf that falls in a temperate forest might take several years to completely decompose, while the same leaf in a tropical rainforest might be processed within months or even weeks. This rapid turnover ensures that nutrients are quickly returned to the system, where they can support new growth, contributing to the remarkable productivity that characterizes rainforest ecosystems. The decomposer's domain operates as a hidden engine that drives the entire rainforest ecosystem, transforming the constant rain of organic matter from the forest canopy into the nutrients that sustain the incredible diversity and productivity we observe above ground. This biological recycling system demonstrates how life has evolved to minimize waste and maximize efficiency, creating closed loops where the death of one organism immediately becomes the foundation for new life, setting the stage for exploring how these recycling processes support the complex movement patterns of forest animals. The rainforest exists not merely as a horizontal landscape, but as a vast three-dimensional realm where movement occurs along vertical pathways as well as terrestrial routes. This complex architecture has given rise to some of nature's most remarkable transportation networks, where animals have evolved sophisticated methods of navigating through multiple layers of forest structure. From the swift highways of the canopy, where primates swing between trees, to the shadowy trails of the forest floor, where ground-dwelling mammals follow scent corridors. These movement patterns reveal how the forest's physical structure both constrains and enables the flow of life through its vertical spaces. The canopy layer serves as the rainforest's primary highway system, supporting a diverse community of animals that rarely descend to the ground. This aerial realm provides continuous pathways formed by interlocking branches that create bridges spanning dozens of meters between tree crowns. The structural complexity of the canopy offers multiple route options at different heights, allowing animals to select pathways based on their size, weight, and locomotory capabilities. Primates represent the most accomplished users of these canopy highways, having evolved a variety of locomotory strategies that exploit different aspects of the forest's three-dimensional structure. Howler monkeys, among the largest arboreal primates, move deliberately through the canopy, using their powerful limbs and prehensile tails to navigate thick branches that can support their considerable weight. Their movements are typically slow and methodical, as they must carefully test each branch before committing their full weight to it. In contrast, spider monkeys have evolved a different approach to canopy travel, specializing in rapid locomotion through smaller branches using their extremely long limbs and hook-like hands. Their lightweight build allows them to access parts of the canopy that would be impossible for heavier primates, while their remarkable reach enables them to span gaps that would stop other species. Spider monkeys demonstrate one of nature's most elegant solutions to three-dimensional travel, moving through the forest with a fluid grace that seems to defy the constraints of gravity. The smaller primates have developed yet another set of adaptations for canopy locomotion. Squirrel monkeys travel in large troops that create living highways through the forest, with individuals following established routes marked by scent and maintained by constant use. Their small size allows them to exploit the finest branches at the canopy's edge, accessing food sources and travel routes unavailable to larger species. Sloths represent perhaps the most specialized approach to arboreal locomotion, moving so slowly and efficiently that they minimize energy expenditure while maximizing their ability to remain undetected by predators. Their powerful claws and unique joint structure allow them to hang beneath branches for extended periods, creating an entirely different type of canopy highway system based on suspension rather than support from above. The structural pathways used by arboreal mammals are not random but follow predictable patterns that reflect both the architecture of the forest and the behavioral needs of the animals. Major highways often develop along the crowns of large emergent trees whose extensive branch systems provide multiple route options. These primary routes may be used by several different species, creating shared pathways that facilitate interactions between different animal communities. Secondary pathways branch off from these major routes, leading to feeding areas, sleeping sites, or territorial boundaries. The development of these pathway systems reflects the cumulative experience of countless individual animals over many generations, with routes being refined and optimized through constant use. Some pathways may be abandoned as forest structure changes through tree falls or growth, while new routes develop as animals explore alternative connections between resource patches. The chemical marking of arboreal pathways creates an additional layer of information that guides animal movement through the canopy. Many primates and other arboreal mammals deposit scent marks along their travel routes, creating chemical maps that provide information about territory boundaries, resource locations, and the recent passage of different individuals. These scent highways can persist for days or weeks, allowing animals to navigate efficiently, even in dense forest where visual landmarks may be limited. Birds exploit canopy highways in fundamentally different ways than mammals, using their flight capabilities to create direct connections between distant parts of the forest. However, many forest bird species still follow predictable flight corridors that take advantage of natural openings in the canopy structure. These aerial highways often follow river valleys, ridge lines, or other landscape features that provide clear flight paths through otherwise dense forest. Mixed species flocking behavior creates temporary highway systems where different bird species travel together through the forest, sharing information about food sources and predator threats. These mobile communities may follow established routes through their territories, creating dynamic pathway networks that change based on seasonal resource availability and breeding requirements. The vertical connectivity between forest layers is maintained by animals that regularly move between canopy and understory environments. Many bird species feed in the canopy but nest in the understory, creating regular vertical traffic patterns that connect different levels of the forest structure. Some mammals, like kinkajous and certain primates, may sleep in the canopy but descend to feed on understory plants or ground-level resources. Lianas and climbing vines provide crucial infrastructure for vertical movement, creating natural ladders that allow animals to move between different forest layers. These plant highways are particularly important for smaller mammals and reptiles that lack the climbing abilities of specialized arboreal species. The three-dimensional network of vines creates alternative pathways that can bypass gaps in the horizontal branch structure of the canopy. The understory layer supports its own network of movement corridors, though these are generally less extensive than canopy highways. The more open structure of the understory allows for both horizontal and vertical movement patterns, with many species using a combination of ground travel and climbing to navigate through this middle layer of the forest. Understory specialists, like certain antbirds, follow army ant columns, creating temporary highway systems that move through the forest as the ants advance through their territory. Ground-level transportation networks in rainforests are characterized by the development of well-defined trail systems that follow the most efficient routes between important resources. These terrestrial highways often develop along natural features like stream valleys, ridge lines, or the bases of large trees where root buttresses create channels for movement. The soft, organic substrate of the forest floor shows clear evidence of repeated use, with compressed soil and vegetation marking the most heavily traveled routes. Large mammals like tapirs and forest elephants create major terrestrial highways that may be used by numerous other species. These well-established trails can persist for decades or even centuries, becoming permanent features of the forest landscape. The regular passage of large animals helps maintain these pathways by preventing vegetation from blocking the roots and by creating scent markers that guide other animals to the established corridors. Smaller ground-dwelling mammals often develop their own network of trails that connect burrows, feeding areas, and other important locations within their territories. These pathways may be invisible to human observers, but are clearly marked by scent and maintained through regular use. The network of small mammal trails creates a fine-scale transportation system that operates beneath the major highways used by larger species. The seasonal dynamics of animal movement create temporal patterns in highway usage that reflect changes in resource availability and breeding requirements. During fruit season, many canopy pathways may experience increased traffic as animals travel longer distances to access productive feeding areas. Conversely, during periods of resource scarcity, movement patterns may become more localized as animals conserve energy and reduce their ranging behavior. Breeding seasons often create dramatic changes in movement patterns as animals travel to traditional nesting or breeding sites. Some species may abandon their normal highway systems entirely during these periods, following ancient routes that connect breeding areas across vast distances. These seasonal highways may only be used for brief periods each year, but represent crucial corridors that maintain genetic connectivity between different forest populations. The impact of tree falls and other natural disturbances on highway systems reveals the dynamic nature of rainforest transportation networks. When large trees fall, they may block established pathways and force animals to develop alternative routes. However, fallen trees also create new opportunities for movement, with their trunks serving as bridges across streams or gaps in the forest structure. Gap formation in the canopy can temporarily disrupt arboreal highway systems, forcing animals to make longer detours or to descend to lower levels to continue their journeys. However, the rapid regeneration of tropical vegetation means that these disruptions are usually temporary, with new growth quickly providing alternative pathways for canopy travel. The role of keystone tree species in maintaining highway connectivity cannot be overstated. Large fig trees, with their extended fruiting seasons and massive crown structures, often serve as major highway intersections where multiple travel routes converge. The loss of these keystone trees can fragment highway systems and force major reorganizations of animal movement patterns. Water features create both barriers and corridors for animal movement through rainforest landscapes. While rivers may block the movement of some arboreal species, they also create travel corridors for semi-aquatic mammals and provide drinking sites that concentrate animal activity. Stream valleys often support their own specialized highway systems, where riparian vegetation creates unique travel opportunities. The chemical ecology of movement corridors involves complex interactions between the scent marks left by different species and the information they provide about territory boundaries, resource locations, and social relationships. Some pathways may be used by multiple species with minimal conflict, while others may be defended territory boundaries that are respected by non-territorial individuals. Nocturnal highway systems often differ dramatically from those used during daylight hours, reflecting the activity patterns of different animal communities. Many rainforest mammals are active only at night, creating transportation networks that operate independently of diurnal systems. These nocturnal highways may follow different routes and connect different resources than those used by day-active species. The maintenance of highway systems requires ongoing effort from the animals that use them. Scent marking must be regularly renewed to maintain chemical signposts, while physical pathways must be kept clear of vegetation growth and debris. This maintenance work represents a significant investment of time and energy that reflects the importance of efficient transportation networks for forest animals. Human activities can severely disrupt natural highway systems, fragmenting pathways and forcing animals to travel longer distances between resources. However, some conservation efforts focus specifically on maintaining corridor connectivity, recognizing that animal movement patterns are essential for ecosystem function and species survival. The evolution of specialized locomotory adaptations reflects millions of years of refinement in response to the three-dimensional challenges of rainforest navigation. Each species has evolved unique solutions to the problems of moving efficiently through complex forest structure while avoiding predators and accessing necessary resources. These intricate movement networks demonstrate how the physical architecture of rainforests enables and shapes animal behavior, creating transportation systems of remarkable complexity that connect all parts of the forest ecosystem. The constant flow of animals along these vertical highways facilitates not only individual survival, but also the broader ecological processes that depend on animal movement, from seed dispersal to nutrient transport, preparing us to explore the sophisticated communication networks that coordinate these complex movements. In the dense, multi-layered world of the rainforest, where visual signals may be blocked by vegetation and sound can be muffled by thick foliage, chemical communication has evolved into one of nature's most sophisticated information systems. Every breath of humid forest air carries thousands of molecular messages, an invisible network of chemical signals that coordinates behaviors, warns of dangers, attracts mates, and maintains the complex relationships that bind the ecosystem together. This chemical language operates continuously, creating a parallel communication network that supplements and often supersedes visual and auditory signals in the management of rainforest life. The chemistry of rainforest communication begins with the unique properties of the tropical atmosphere itself. The warm, humid conditions that characterize these environments create ideal circumstances for the dispersal and detection of chemical signals. High humidity keeps volatile organic compounds suspended in the air longer than they would persist in drier environments, while the still air conditions beneath the forest canopy allow chemical gradients to develop and maintain their integrity over considerable distances. The molecular diversity of chemical signals in rainforests reflects the extraordinary biological diversity of these ecosystems. Each species has evolved its own unique chemical signature, a combination of compounds that serves as its identification card in the forest's molecular communication network. These chemical signatures are often species-specific and sometimes even individual-specific, allowing for remarkably precise communication in environments where dozens or hundreds of species may coexist in close proximity. Plant chemical communication represents one of the most complex aspects of rainforest molecular messaging, with plants using chemical signals to coordinate numerous essential functions. When attacked by herbivorous insects, many rainforest plants release volatile compounds that serve multiple communication functions simultaneously. These stress-induced chemicals can attract predatory insects that will attack the herbivores, effectively calling for help in the plant's defense. The same chemicals may also serve as warning signals to neighboring plants, triggering them to begin producing defensive compounds before they are actually attacked. The specificity of plant chemical signaling is remarkable in its precision. Different types of herbivore damage can trigger the release of different chemical signals, allowing plants to summon specific types of predators or parasites that specialize in controlling particular pest species. A plant attacked by caterpillars might release compounds that attract parasitic wasps, while the same plant attacked by aphids could produce different chemicals that draw predatory beetles or other aphid specialists. The temporal dynamics of plant chemical communication create complex patterns of molecular messaging that change throughout the day and across seasons. Many plants release different chemical signals during different parts of their daily cycles, with some compounds being produced primarily during daylight hours when photosynthesis is active, while others are released at night when different types of interactions are more likely to occur. These temporal patterns help coordinate plant responses with the activity cycles of their partners and antagonists. Mycorrhizal networks facilitate underground chemical communication between plants, allowing trees and other forest plants to share information about resource availability, pathogen threats, and environmental stresses. These fungal networks can transport both nutrients and chemical signals between widely separated plants, creating what some researchers describe as a plant communication internet that spans entire forest areas. Through these networks, established trees can chemically communicate with seedlings, potentially sharing information that improves the survival chances of young plants. The chemical ecology of plant reproduction involves some of the most sophisticated molecular signaling systems found in nature. Flowering plants produce complex bouquets of volatile compounds designed to attract specific pollinators, while sometimes simultaneously repelling organisms that might damage flowers without providing pollination services. The chemical composition of these floral scents can be incredibly specific, with some orchids producing scent profiles that mimic the pheromones of female insects so precisely that male insects are deceived into attempting to mate with the flowers. The timing of floral scent production is often coordinated with the activity patterns of specific pollinators, with some flowers releasing their strongest chemical signals during the times of day when their preferred pollinators are most active. Night-blooming flowers may produce intense scents that can attract moths and bats from considerable distances, while day-blooming species might produce different chemical signals optimized for bee or bird pollinators. Animal pheromone systems in rainforests demonstrate remarkable complexity and specificity in their molecular messaging capabilities. Insects, with their highly sensitive chemoreceptor systems, have evolved some of the most sophisticated chemical communication networks found anywhere in nature. Social insects like ants and termites use chemical signals to coordinate virtually every aspect of their colony activities, from foraging and nest construction to defense and reproduction. Ant chemical communication systems involve multiple types of pheromones that convey different types of information to colony members. Trail pheromones mark efficient pathways between the nest and food sources, with the chemical concentration indicating both the direction of travel and the quality of the resources at the trail's end. Alarm pheromones alert colony members to threats, while different chemical signals coordinate different types of defensive responses depending on the nature of the threat. The sophistication of ant chemical communication extends to the ability to combine different pheromone signals to create complex messages. Ants may use combinations of chemicals to indicate not just the presence of food, but also its type, quality, and distance from the nest. Some species can even use chemical gradients to provide directional information, allowing colony members to follow chemical trails with remarkable precision, even in the complete darkness of underground nests. Termite colonies employ similarly complex chemical communication systems, with different castes of individuals producing different types of pheromone signals. Queen termites produce pheromones that regulate the development and behavior of other colony members, while worker termites use chemical signals to coordinate construction activities and food processing operations. Soldier termites may produce alarm pheromones that trigger defensive responses throughout the colony when threats are detected. Mammalian scent communication in rainforests involves both volatile compounds that can be detected at a distance, and non-volatile chemicals that require direct contact for detection. Many rainforest mammals maintain scent marking systems that provide information about territory boundaries, reproductive status, and individual identity. These chemical markers can persist for days or weeks in the humid forest environment, creating relatively permanent information systems that operate independently of the animals that created them. The complexity of mammalian scent signals often involves combinations of dozens or even hundreds of different chemical compounds, creating unique molecular signatures that can convey multiple types of information simultaneously. A single scent mark might indicate the species, sex, age, reproductive status, health condition, and individual identity of the animal that produced it, along with information about when the mark was made and potentially what the animal had been eating recently. Primate chemical communication demonstrates particular sophistication in rainforest environments where visual signals may be limited by dense vegetation. Many primate species use scent marking to establish territory boundaries and communicate social status, with different chemical compounds conveying different types of social information. Some primates can recognize individual group members by scent alone, while others use chemical signals to coordinate group movements and maintain social cohesion in dense forest environments. The chemical communication systems used by rainforest birds often involve less obvious molecular signaling than those of mammals or insects, but recent research has revealed that many bird species do produce and respond to chemical signals. Some birds use scent to recognize nest sites or to identify appropriate nesting materials, while others may use chemical cues to assess the quality of potential mates or territory sites. Reptile and amphibian chemical communication systems are particularly well developed in rainforest environments, where these animals must often rely on chemical cues to locate mates and suitable habitat. Many tropical frogs use chemical signals to identify breeding sites and to communicate with potential mates, while some species produce chemical compounds that help them recognize members of their own species in environments where multiple closely related species may occur together. The integration of chemical communication systems across different taxonomic groups creates complex networks of molecular information exchange that help coordinate ecosystem-level processes. The chemical signals produced by plants may influence the behavior of insects, which in turn affects the chemical signals produced by other plants, creating feedback loops that help maintain ecological stability and coordinate responses to environmental changes. Seasonal patterns in chemical communication reflect the changing needs and activities of rainforest organisms throughout the year. During reproductive seasons, many species increase their production of sex pheromones and other chemical signals related to mating behavior, creating periods of intensified chemical communication activity. Similarly, during periods of resource scarcity or environmental stress, many organisms may alter their chemical signaling patterns to coordinate adaptive responses. The role of chemical communication in predator-prey interactions adds another layer of complexity to rainforest molecular messaging systems. Predators may use chemical cues to locate prey, while prey species often produce alarm chemicals when threatened that warn other individuals of danger. Some prey species have evolved the ability to detect and interpret the chemical signals produced by their predators, allowing them to avoid dangerous areas or times when predation risk is highest. The evolution of chemical resistance and counter-adaptations creates ongoing arms races between different species that communicate chemically. Plants may evolve new defensive chemicals in response to herbivore adaptations, while herbivores develop increased resistance to plant toxins. Similarly, prey species may evolve reduced chemical signatures that make them harder for predators to detect, while predators develop increased sensitivity to faint chemical cues. Human activities can disrupt natural chemical communication systems in various ways, from air pollution that interferes with chemical signal transmission to habitat fragmentation that breaks up the spatial scales over which chemical communication normally operates. Understanding these impacts is crucial for conservation efforts, as the disruption of chemical communication networks can have cascading effects throughout entire ecosystems. The study of chemical communication in rainforests continues to reveal new levels of complexity and sophistication in these molecular messaging systems. Advanced analytical techniques are allowing researchers to identify and characterize chemical signals that were previously undetectable, while behavioral studies are revealing how organisms integrate chemical information with other types of sensory input to make complex decisions about feeding, reproduction, and social behavior. The chemical communication networks of rainforest ecosystems demonstrate how molecular information systems can coordinate biological processes across multiple scales, from individual organisms to entire ecosystems. These invisible networks of chemical signals help maintain the stability and resilience of rainforest communities by facilitating the rapid transmission of information about environmental changes, resource availability, and threats, preparing the ground for understanding how these communication systems contribute to the forest's role as an evolutionary laboratory where new species continuously arise. Rainforests function as Earth's greatest laboratories for evolutionary innovation, generating new species at rates that exceed those of any other terrestrial ecosystem. The extraordinary diversity that characterizes tropical forests is not merely the result of species accumulating over time, but rather reflects ongoing processes of evolutionary change that continue to produce new forms of life. The complex three-dimensional structure, stable climate, and intricate web of ecological relationships create conditions that both promote the evolution of new species and allow countless specialized forms to coexist in remarkably small areas. The process of speciation, the evolutionary development of new species from existing ones, requires particular conditions that rainforests provide in abundance. Geographic isolation, reproductive barriers, and ecological opportunities must align to allow populations to diverge genetically until they become distinct species. Rainforests offer multiple mechanisms for creating the isolation necessary for speciation while simultaneously providing the stable environmental conditions that allow evolutionary processes to proceed without constant disruption from environmental catastrophes. The complex topography of many rainforest regions creates natural barriers that can isolate populations and promote speciation. River systems carve deep valleys through forest landscapes, creating barriers that may be insurmountable for some species while remaining permeable to others. Over geological timescales, the meandering of rivers and the formation of new waterways can repeatedly subdivide populations, creating opportunities for independent evolutionary trajectories. Mountain ridges, valley systems, and even large fallen trees can serve as barriers that promote population isolation and subsequent speciation. Within the forest itself, the vertical stratification creates numerous microhabitats that can support independent evolutionary processes. Species adapted to life in the canopy may become reproductively isolated from closely related species living in the understory, even when both populations exist in the same geographic location. This habitat-based isolation can be just as effective as geographic separation in promoting speciation, particularly when different forest layers provide distinct selective pressures that favor different adaptations. The extraordinary structural complexity of rainforest environments creates countless ecological niches that can support highly specialized species. Each combination of height, light level, moisture, temperature, and biotic interactions represents a potential niche that evolution can fill with appropriately adapted organisms. The three-dimensional nature of rainforest structure multiplies the number of available niches compared to simpler ecosystems, providing opportunities for adaptive radiation where single ancestral species give rise to multiple descendant species adapted to different aspects of the forest environment. Adaptive radiation in rainforests often occurs when colonizing species encounter environments with numerous unfilled ecological niches. Islands and isolated forest patches frequently demonstrate this process, with founding species rapidly diversifying to exploit different resources or habitats available in their new environment. The famous adaptive radiations of Darwin's finches in the Galapagos or the cichlid fish in African lakes find their terrestrial equivalent in the diversification of plant and animal lineages within rainforest environments. The remarkable diversity of epiphytic plants illustrates how rainforest structure promotes evolutionary innovation. The three-dimensional branching architecture of forest trees creates thousands of distinct microhabitats in the canopy, each with slightly different conditions of light, moisture, nutrients, and physical support. Epiphytic species have evolved to exploit virtually every possible niche in this aerial environment, from the sun-baked outer branches of emergent trees to the perpetually moist crevices in the inner canopy. Different types of epiphytic plants have evolved distinct strategies for surviving in their aerial habitats, leading to extraordinary diversification within groups like orchids, bromeliads, and ferns. Some species have evolved thick, succulent leaves for water storage, while others have developed specialized root systems for capturing nutrients from the air. Still others have formed partnerships with ants or other animals, creating entirely new types of ecological relationships that would be impossible in simpler environments. The stability of rainforest climates over geological timescales has been crucial in allowing evolutionary processes to proceed without constant environmental disruption. Unlike temperate regions that have experienced repeated ice ages and dramatic climate fluctuations, many tropical regions have maintained relatively stable conditions for millions of years. This stability has provided the time necessary for complex evolutionary processes to unfold, allowing for the gradual accumulation of species and the development of intricate ecological relationships. However, this stability has not been absolute, and periodic environmental changes have provided additional opportunities for speciation. Climate fluctuations during ice ages affected even tropical regions, causing forest distributions to shift and creating periods of isolation and reconnection that promoted evolutionary divergence. The expansion and contraction of forest areas created refugia, where species could persist during unfavorable periods, while subsequent forest expansion allowed for recolonization and the establishment of new evolutionary trajectories. The role of pollinators and seed dispersers in promoting plant speciation cannot be overstated. The extraordinary diversity of animal pollinators in rainforests has allowed for the evolution of highly specialized plant-pollinator relationships that can lead to reproductive isolation and speciation. When plant populations become specialized for pollination by different animal species, they may become reproductively isolated even when growing in close proximity, allowing for sympatric speciation where new species arise without geographic separation. The diversity of seed dispersal mechanisms in rainforests similarly promotes plant speciation by creating opportunities for population isolation and the colonization of new habitats. Plants dispersed by different animal species may colonize different parts of the forest landscape, leading to geographic isolation that promotes evolutionary divergence. The complex relationships between plants and their animal dispersers can also create selective pressures that drive the evolution of new species adapted to particular dispersal partnerships. Sexual selection has played a particularly important role in animal speciation within rainforest environments. The complex three-dimensional structure of forests provides numerous opportunities for the evolution of elaborate courtship displays and sexual signals that can lead to reproductive isolation and speciation. Bird species may evolve different song patterns adapted to the acoustic properties of different forest microhabitats, while the visual displays of insects and other animals may become specialized for the light conditions of particular forest layers. The evolution of mimicry systems in rainforests demonstrates how complex ecological interactions can drive speciation. When multiple species evolve to share similar warning signals, or when harmless species evolve to mimic dangerous ones, the resulting selective pressures can promote the diversification of both model and mimic species. These mimicry complexes often involve multiple species and can create evolutionary arms races that drive continued speciation and diversification. Parasite-host relationships provide another engine for evolutionary change and speciation in rainforest environments. The extraordinary diversity of hosts in tropical forests supports equally diverse parasite communities, and the coevolutionary interactions between parasites and hosts can drive rapid evolutionary change in both groups. Host switching by parasites can lead to the evolution of new parasite species, while hosts evolve defensive strategies that may promote their own diversification. The phenomenon of ecological release occurs when species colonize new environments where they face reduced competition or predation pressure. In rainforest environments, this can happen when species colonize new habitat types or when environmental changes create new ecological opportunities. Ecological release can lead to adaptive radiation as species expand their ecological roles and diversify to exploit newly available resources. Chemical diversity in rainforest plants provides both the raw material for evolutionary innovation and the selective pressures that drive continued diversification. Plants have evolved thousands of different secondary compounds that serve as defenses against herbivores, pathogens, and competitors. The arms race between plants and their antagonists has driven the evolution of increasingly sophisticated chemical defenses, while herbivores and pathogens have evolved counter-adaptations that allow them to overcome these defenses. The evolution of complex life cycles in rainforest animals demonstrates how environmental complexity can promote evolutionary innovation. Many rainforest species have evolved life cycles that exploit different resources or habitats at different life stages, reducing competition and allowing for greater specialization. These complex life cycles often require precise timing and coordination with environmental conditions, creating selective pressures that can lead to reproductive isolation and speciation. Hybridization between related species can actually promote speciation in some circumstances, particularly in rainforest environments, where complex ecological interactions create opportunities for the establishment of hybrid populations. When hybrids occupy ecological niches different from either parent species, they may evolve into distinct species through a process called hybrid speciation. The role of genetic drift in small populations has been particularly important in promoting speciation in fragmented rainforest landscapes. When populations become small due to habitat fragmentation or other factors, random genetic changes can become fixed in populations, leading to evolutionary divergence even in the absence of strong selective pressures. This process has likely contributed to the evolution of many endemic species in isolated forest fragments. Molecular genetic techniques have revealed that cryptic speciation, the evolution of genetically distinct species that are morphologically similar, is much more common in rainforests than previously recognized. Many groups of organisms that were thought to represent single widespread species have been shown to actually consist of multiple closely related species that have diverged genetically while retaining similar morphological characteristics. The concept of evolutionary potential describes the capacity of populations and ecosystems to continue evolving and adapting to changing conditions. Rainforests maintain high evolutionary potential through their genetic diversity, population connectivity, and environmental heterogeneity. This evolutionary potential is crucial for the long-term persistence of rainforest ecosystems in the face of ongoing environmental changes. The feedback loops between diversity and diversification mean that high species diversity itself promotes further evolutionary innovation. Diverse communities create more complex ecological networks with more potential interactions, which in turn create more opportunities for coevolution and specialization. This positive feedback helps explain why rainforests continue to generate new species even after millions of years of evolutionary history. Conservation genetics has revealed that maintaining evolutionary processes requires preserving not just individual species, but the ecological contexts and evolutionary opportunities that allow speciation to continue. This understanding has important implications for rainforest conservation, suggesting that protecting the connected forest areas is crucial for maintaining the evolutionary processes that generate and maintain biodiversity. The study of evolutionary ecology in rainforests continues to reveal new mechanisms of speciation and new examples of ongoing evolutionary change. These discoveries demonstrate that rainforests are not static repositories of ancient diversity, but rather dynamic systems where evolution continues to create new forms of life, establishing the foundation for understanding how this remarkable diversity influences the seasonal rhythms that coordinate forest life. To the casual observer, tropical rainforests might appear to exist in a world without seasons, where the endless summer of warm temperatures and abundant rainfall creates conditions of perpetual sameness. Yet beneath this apparent uniformity lies a subtle but profound seasonal rhythm that orchestrates one of nature's most complex symphonies of timing and coordination. The seasonal changes in rainforests may be less dramatic than the stark contrasts between winter and summer in temperate regions, but they are no less important in shaping the lives and behaviors of the countless species that call these forests home. The seasonal patterns of tropical rainforests are defined primarily by changes in rainfall rather than temperature, creating wet and dry seasons that profoundly influence every aspect of forest ecology. These precipitation patterns reflect the movement of massive weather systems, like the Intertropical Convergence Zone, where trade winds from the northern and southern hemispheres meet and create conditions for intense rainfall. As this zone shifts throughout the year, it brings periods of heavy rain, followed by relatively drier months, creating the fundamental rhythm that drives seasonal changes in tropical forests. The transition between wet and dry seasons is often gradual rather than abrupt, with rainfall patterns shifting over periods of weeks or months. During the height of the wet season, daily afternoon thunderstorms may deliver torrential downpours that can drop several centimeters of rain in just a few hours. These intense precipitation events flood temporary streams, saturate soils, and maintain the high humidity levels that many forest organisms depend upon for their survival. As the dry season approaches, the frequency and intensity of rainfall gradually diminish, though even the driest months in most rainforest regions receive more precipitation than many temperate areas see in their wettest seasons. The reduction in rainfall during dry periods can still have dramatic effects on forest ecosystems, as many biological processes are finely tuned to the subtle variations in water availability that characterize seasonal transitions. Plant reproductive cycles demonstrate remarkable coordination with these seasonal rainfall patterns, with many species timing their flowering and fruiting to coincide with optimal conditions for pollination, seed development, and dispersal. The onset of the wet season often triggers synchronized flowering events across multiple tree species, creating spectacular displays of color and fragrance that attract pollinators from throughout the forest. This timing ensures that flowers have access to abundant water during the energy-intensive process of producing nectar and maintaining blooms. Tree species that rely on wind dispersal for their seeds often time their reproduction to coincide with the transition between seasons, when changing weather patterns create the air currents necessary for effective seed transport. The beginning of the dry season may bring increased winds that can carry winged seeds across great distances, while the approach of the wet season provides the moisture that seeds need for successful germination and establishment. Fruiting patterns in rainforest trees show equally sophisticated timing mechanisms that coordinate with both seasonal conditions and the life cycles of animal dispersers. Many large-seeded tree species produce their fruits during the wet season, when abundant rainfall supports rapid germination and seedling growth. The timing of fruit production also reflects the seasonal movements and reproductive cycles of the birds and mammals that serve as seed dispersers, ensuring that fruits are available when disperser populations are most active and mobile. The phenomenon of massed fruiting, where multiple tree species produce synchronized crops of fruits or seeds, demonstrates how seasonal cues can coordinate reproductive timing across entire forest communities. These mass fruiting events often follow subtle environmental triggers like slight changes in day length, temperature fluctuations, or rainfall patterns that signal optimal conditions for reproduction. The synchronization benefits individual trees by overwhelming seed predators and ensuring that at least some seeds escape consumption and successfully establish new populations. Animal reproductive cycles in rainforests show equally precise coordination with seasonal patterns, though the cues they respond to may be different from those that influence plants. Many mammals time their breeding to ensure that offspring are born during periods when food resources are most abundant, often coordinating with the fruiting cycles of the plants they depend upon for nutrition. The gestation periods of many tropical mammals appear to have evolved to synchronize birth timing with seasonal peaks in resource availability. Bird breeding seasons frequently coincide with the transition from dry to wet seasons, when increasing rainfall stimulates insect activity and plant growth that provide abundant food for feeding nestlings. The timing of breeding also reflects the availability of suitable nesting materials and sites, with many species beginning construction activities as new plant growth provides fresh materials for nest building. The seasonal movements of many rainforest animals reflect their responses to changing patterns of resource availability throughout the year. While true migration is less common in tropical forests than in temperate regions, many species undertake regular seasonal movements that track the availability of food resources or suitable habitat conditions. These movements may involve vertical migration within the forest structure, with some species spending different seasons at different heights in the forest canopy. Fruit-eating birds and mammals often follow predictable seasonal routes that connect areas where different tree species fruit at different times of year. These frugivorous species may travel considerable distances to track the seasonal progression of fruit availability, creating temporary concentrations of animals that can dramatically affect local ecological processes. The movements of these mobile species help connect different areas of the forest through their role in seed dispersal and nutrient transport. The seasonal activity patterns of insects demonstrate some of the most precisely timed responses to environmental cues found in rainforest ecosystems. Many insect species coordinate their life cycles with specific seasonal conditions, emerging as adults during periods when food resources are abundant or when weather conditions favor reproduction and dispersal. The mass emergence of certain insect species can create seasonal pulses of food availability that influence the behavior and distribution of insectivorous birds and other predators. Butterfly and moth populations often show dramatic seasonal fluctuations that reflect both the availability of host plants for their larvae and the weather conditions that affect adult activity. Some species time their emergence to coincide with the blooming of specific plants that provide nectar for adults, while others synchronize their reproduction with the seasonal growth of plants that serve as food for their caterpillars. The timing of decomposition processes shows seasonal variation that reflects changes in temperature, moisture, and the availability of organic matter. During the wet season, high temperatures and abundant moisture accelerate decomposition rates, rapidly processing the organic matter that accumulates on the forest floor. The increased activity of decomposer organisms during wet periods helps ensure that nutrients are released when plants are most active and capable of absorbing them. Dry season conditions may slow some decomposition processes while favoring others, creating temporal patterns in nutrient availability that many forest organisms have evolved to exploit. The reduced moisture during dry periods can concentrate nutrients in smaller volumes of soil solution, potentially making them more readily available to plant roots that have evolved to take advantage of these seasonal concentrations. The seasonal dynamics of mycorrhizal fungi reflect their role in facilitating nutrient exchange between plants and their environment. Many mycorrhizal species show increased activity during transitions between seasons when changing environmental conditions create opportunities for establishing new partnerships with plant roots or expanding existing fungal networks. The timing of spore production and dispersal in many fungal species corresponds to seasonal patterns in rainfall and temperature that favor successful establishment of new fungal colonies. Pollination networks demonstrate remarkable seasonal organization, with different plant species flowering at different times throughout the year to minimize competition for pollinators while ensuring adequate pollination services. This temporal partitioning of resources allows numerous plant species to coexist in the same area by avoiding direct competition for the limited attention of available pollinators. Some pollinator species have evolved life cycles that track the seasonal progression of flowering plants, timing their own reproduction and activity patterns to coincide with periods of maximum floral resource availability. Specialized pollinators may even undergo seasonal migrations or dormancy periods that align with the flowering schedules of their preferred plant partners. The chemical ecology of seasonal timing involves complex interactions between environmental cues and the physiological mechanisms that control reproductive timing and other life cycle events. Plants and animals use various environmental signals to assess seasonal conditions and coordinate their activities accordingly. Changes in day length, temperature fluctuations, rainfall patterns, and even subtle variations in humidity can serve as seasonal cues that trigger specific behavioral or physiological responses. Hormonal systems in both plants and animals integrate multiple environmental cues to produce coordinated responses to seasonal changes. These endocrine mechanisms allow organisms to anticipate seasonal transitions and begin preparing for changing conditions before they actually occur. The precision of these timing systems reflects millions of years of evolutionary refinement in response to the predictable seasonal patterns that characterize tropical forest environments. The social coordination of seasonal activities demonstrates how temporal organization can extend beyond individual organisms to encompass entire communities. Many social insects coordinate their colony activities with seasonal patterns, timing the production of reproductive individuals to coincide with optimal conditions for dispersal and new colony establishment. The seasonal activities of social species can have cascading effects on other forest organisms that depend on their ecological services. Predator-prey relationships show seasonal fluctuations that reflect the changing abundance and activity patterns of both predators and their prey. Seasonal peaks in prey availability may support increased predator reproduction, while periods of prey scarcity may force predators to alter their foraging strategies or territory sizes. These seasonal dynamics help maintain the balance between predator and prey populations over annual cycles. The phenology of rainforest ecosystems, the study of seasonal timing and biological events, reveals intricate networks of coordination that connect species across multiple trophic levels. The timing of one species' reproductive cycle may influence the seasonal activities of numerous other species through direct and indirect ecological interactions. Understanding these phenological networks is crucial for predicting how rainforest ecosystems might respond to climate change and other environmental perturbations. Climate change poses particular challenges for the seasonal coordination systems that have evolved in rainforest ecosystems over thousands of years. Shifts in the timing or intensity of seasonal rainfall patterns could disrupt the precisely timed reproductive cycles and ecological interactions that depend on predictable seasonal cues. Some species may be able to adjust their timing in response to changing conditions, while others may find their life cycles increasingly out of synchrony with their environment. The conservation implications of seasonal timing mechanisms highlight the importance of maintaining large, intact forest areas that can support the full range of seasonal movements and activities that many rainforest species require. Habitat fragmentation can disrupt seasonal migration patterns and eliminate the seasonal resource patches that many species depend upon for successful reproduction and survival. These intricate seasonal rhythms reveal how even the most stable ecosystems on Earth are governed by temporal patterns that organize and coordinate the activities of countless species, creating a foundation of predictability that allows complex ecological relationships to persist and flourish across time, preparing us to explore the remarkable adaptations that have evolved in response to these seasonal and environmental challenges. The rainforest stands as nature's greatest showcase of evolutionary ingenuity, where millions of years of natural selection have crafted solutions to biological challenges that seem to defy the limits of possibility. Each organism in these complex ecosystems carries within its anatomy, physiology, and behavior a collection of adaptations so precisely tuned to their environment that they represent masterpieces of evolutionary engineering. From microscopic cellular modifications to elaborate morphological structures, these adaptations reveal how the demanding conditions and abundant opportunities of rainforest life have driven the evolution of some of nature's most extraordinary innovations. The three-dimensional complexity of rainforest environments has created unique challenges for locomotion that have driven the evolution of remarkable movement adaptations. Tree frogs exemplify this evolutionary response with their specialized toe pads, structures that represent one of nature's most elegant solutions to the problem of moving through vertical environments. These adhesive pads contain millions of microscopic hairs called setae that interact with surface molecules through van der Waals forces, allowing frogs to cling to smooth leaves and wet bark with remarkable tenacity. The engineering principles behind tree frog adhesion demonstrate sophisticated biomechanical solutions that human technology is only beginning to understand. The toe pads can support many times the frog's body weight while remaining reversible, allowing the animal to detach and move with fluid grace through the canopy. The pads work effectively on both rough and smooth surfaces in wet and dry conditions, making them remarkably versatile tools for arboreal life. Different species of tree frogs have evolved variations in their adhesive systems that reflect their specific locomotory needs and habitat preferences. Species that primarily inhabit smooth-barked trees may have toe pads with different microscopic structures than those that prefer rough surfaces, while frogs that are active in wet conditions have evolved pads that maintain their effectiveness even when covered with moisture. The remarkable climbing abilities of rainforest mammals showcase another approach to three-dimensional locomotion. Primates have evolved diverse solutions to the challenges of arboreal movement, with each major group developing distinct adaptations that allow them to exploit different aspects of the forest's vertical structure. Spider monkeys have evolved extremely long limbs and hook-shaped hands that allow them to swing through the canopy with remarkable speed and efficiency, using a form of locomotion called brachiation that is essentially aerial acrobatics performed dozens of meters above the ground. The prehensile tails of many New World primates represent another evolutionary innovation that has enhanced their three-dimensional mobility. These tails function as fifth limbs, capable of supporting the animal's full weight while leaving the hands and feet free for other activities. The tip of a prehensile tail contains the same types of specialized skin found on primate fingertips, complete with dermal ridges that improve grip on smooth surfaces. Sloths have evolved perhaps the most energy-efficient approach to arboreal locomotion, moving so slowly and deliberately that they minimize the energy costs of three-dimensional travel. Their specialized claws are permanently curved and lock into position around branches, allowing sloths to hang motionless for hours without muscular effort. This adaptation enables them to remain virtually invisible to predators while conserving energy in an environment where food sources may be widely scattered and low in nutritional value. The remarkable diversity of bird beaks in rainforest environments demonstrates how morphological adaptations can be fine-tuned to exploit specific food resources. Hummingbird beaks show extraordinary variation in length, curvature, and shape, with each species specialized for feeding on particular types of flowers. Some species have evolved curved beaks that perfectly match the curvature of specific flowers, creating lock and key relationships between bird and plant that benefit both partners. Toucan beaks represent a different type of evolutionary solution, combining lightweight construction with remarkable strength and versatility. Despite their impressive size, these beaks are surprisingly light due to their internal honeycomb structure, which provides strength while minimizing weight. The large surface area of the beak also serves as a thermal regulation system, helping toucans maintain optimal body temperature in warm tropical environments. The specialized feeding adaptations of rainforest birds extend beyond beak morphology to include behavioral and physiological innovations. Some species have evolved the ability to process toxic fruits that would be harmful to other animals, allowing them to exploit food sources that are unavailable to competitors. These detoxification abilities often involve specialized liver enzymes or other physiological mechanisms that neutralize plant secondary compounds. Insect adaptations in rainforest environments showcase some of evolution's most intricate solutions to the challenges of survival in complex ecosystems. Stick insects have evolved morphological and behavioral adaptations that make them virtually indistinguishable from the twigs and branches on which they rest. Their body proportions, coloration, and even surface texture perfectly mimic plant structures, while their behavior includes swaying movements that simulate the motion of vegetation in gentle breezes. Butterfly wing patterns demonstrate how visual adaptations can serve multiple functions simultaneously. The eye spots on many butterfly wings can startle predators by mimicking the eyes of larger animals, while other wing patterns provide camouflage that makes butterflies nearly invisible when their wings are folded. Some species have evolved transparent wings that make them difficult to detect as they fly through dappled forest light. The evolution of complex life cycles in rainforest insects represents another type of adaptive innovation that allows species to exploit different resources and avoid predators at different life stages. Many tropical insects have larvae that live in completely different environments and feed on entirely different resources than the adults, effectively allowing a single species to occupy multiple ecological niches. Plant adaptations in rainforest environments reveal solutions to the unique challenges of photosynthesis, water management, and reproduction in tropical conditions. Epiphytic plants have evolved remarkable water collection and storage systems that allow them to thrive without soil contact. Tank bromeliads create their own water reservoirs using specially modified leaves, while other epiphytes have developed water-absorbing scales or specialized root systems that can extract moisture directly from humid air. The leaves of many rainforest plants show adaptations to the challenges of maintaining photosynthesis while managing water loss and avoiding damage from intense rainfall. Drip tips on leaves facilitate water removal and prevent the growth of harmful fungi, while specialized leaf surfaces may have microscopic structures that enhance light capture or reduce water loss through transpiration. The extraordinary chemical diversity of rainforest plants represents one of evolution's most sophisticated adaptive responses to herbivory and pathogen pressure. Plants have evolved thousands of different secondary compounds that serve as chemical defenses against insects, fungi, and other organisms that might harm them. These chemical adaptations often show remarkable specificity, with different compounds being effective against different types of threats. Some plants have evolved inducible defenses that are activated only when the plant is actually under attack, allowing them to minimize the energy costs of defense while maintaining protection when needed. These systems can respond rapidly to herbivore damage, producing defensive compounds within hours of an attack, and sometimes even warning neighboring plants through chemical signals. Root adaptations in rainforest plants reflect the challenges of obtaining nutrients from poor tropical soils while coping with periods of waterlogging and drought. Many trees have evolved extensive shallow root systems that maximize their ability to capture nutrients from decomposing organic matter on the forest floor. Buttress roots provide structural support while creating catchment areas where organic matter can accumulate and be processed by the tree's surface roots. Some plants have evolved specialized root structures called pneumatophores that can absorb oxygen from the air when soils become waterlogged. These aerial roots allow plants to continue respiration even when their main root systems are submerged, providing crucial flexibility in environments where water levels can fluctuate dramatically. The reproductive adaptations of rainforest organisms showcase evolution's solutions to the challenges of finding mates and reproducing successfully in complex environments. Many plants have evolved elaborate flower structures and chemical attractants that ensure pollination by specific animal partners, while some have developed reproductive strategies that reduce their dependence on animal pollinators entirely. The timing of reproductive activities often shows precise coordination with environmental conditions, with many species having evolved internal biological clocks that coordinate reproduction with optimal conditions for offspring survival. These timing mechanisms may respond to subtle environmental cues that are barely detectable to human observers, but provide reliable signals about seasonal conditions to the organisms that have evolved to use them. Seed dispersal adaptations demonstrate how plants have evolved to exploit the movements and behaviors of animal dispersers. Some seeds have evolved hooks or barbs that attach to animal fur or feathers, while others are packaged in nutritious fruits that encourage consumption and transport by frugivorous animals. The timing of fruit production and the chemical composition of fruits often show precise coordination with the life cycles and preferences of disperser species. Defensive adaptations in rainforest animals range from passive camouflage to active chemical defenses that can deter or harm potential predators. Poison dart frogs have evolved some of nature's most potent toxins, which they acquire from their diet of alkaloid-rich insects and concentrate in specialized skin glands. Their bright warning coloration advertises their toxicity to potential predators, creating an effective deterrent system that protects both the frogs and any predators that might otherwise waste energy attacking them. Many rainforest animals have evolved behavioral adaptations that complement their morphological and physiological specializations. Mixed species foraging flocks demonstrate how different bird species have evolved complementary foraging strategies that benefit all participants. Some species specialize in detecting and alerting others to predator threats, while others excel at finding food sources that can be shared among flock members. The social adaptations of many rainforest species reflect the advantages of cooperation in complex environments where resources may be widely scattered and threats can come from multiple directions. Army ant colonies demonstrate how social organization can create superorganisms capable of overwhelming prey much larger than individual ants. While leafcutter ants have evolved agricultural systems that allow them to cultivate food in environments where suitable nutrition would otherwise be scarce. Sensory adaptations in rainforest animals often reflect the challenges of communication and navigation in dense, multi-layered environments. Many species have evolved enhanced chemical senses that allow them to detect and interpret the complex bouquet of scents that characterize forest environments. Others have developed specialized acoustic abilities that allow them to communicate effectively in environments where sound transmission may be limited by vegetation. The remarkable diversity of adaptations found in rainforest ecosystems reflects not only the challenges these environments present, but also the extraordinary opportunities they provide for evolutionary innovation. The complex three-dimensional structure, stable climate, and intricate web of ecological relationships create conditions where highly specialized adaptations can evolve and persist, leading to levels of biological diversity and ecological complexity found nowhere else on Earth. These adaptations collectively demonstrate how evolutionary processes can produce solutions of remarkable elegance and efficiency when given sufficient time and appropriate environmental conditions. Each adaptation represents a successful solution to specific environmental challenges, while simultaneously creating new opportunities and challenges that drive further evolutionary innovation. The continuing evolution of new adaptations in rainforest environments ensures that these ecosystems remain dynamic and responsive to changing conditions, maintaining the resilience and stability that allow them to persist as complex, integrated systems where countless specialized organisms work together to maintain the delicate balance that sustains all life, preparing us to examine how these intricate relationships maintain ecosystem stability through the complex dynamics of predation. In the intricate web of rainforest life, the relationship between predators and their prey forms one of nature's most fundamental organizing principles, creating a dynamic balance that maintains the health and diversity of entire ecosystems. Far from being merely a struggle between hunter and hunted, these relationships represent sophisticated evolutionary partnerships that have shaped the behavior, morphology, and ecology of countless species over millions of years. The complex three-dimensional structure of rainforests provides an elaborate stage upon which these interactions unfold, offering countless opportunities for both hunting success and escape while driving continuous evolutionary innovation in both predators and their prey. The architectural complexity of rainforests creates hunting environments unlike those found in any other terrestrial ecosystem. The multiple layers of vegetation provide predators with numerous ambush sites, from the shadowy understory where jaguars stalk through dappled light to the dense canopy where arboreal snakes wait motionlessly along branch highways used by unsuspecting primates. This three-dimensional hunting arena requires predators to develop specialized strategies for each forest layer, while prey species must remain vigilant for threats that can approach from above, below, or any horizontal direction. Arboreal predators have evolved remarkable adaptations for hunting in three-dimensional space. Tree-dwelling cats like margays and ocelots possess enhanced balance and climbing abilities that allow them to pursue prey along even the thinnest branches. Their semi-retractable claws provide exceptional grip on bark and branches, while their long tails serve as dynamic stabilizers that can adjust position rapidly during pursuit. These cats have even evolved the ability to rotate their hind feet 180 degrees, allowing them to descend trees headfirst, like squirrels, a unique adaptation among felines that reflects the demands of their arboreal hunting lifestyle. The hunting strategies of rainforest predators often involve remarkable patience and precise timing. Harpy eagles, among the most powerful raptors in the world, may spend hours perched motionlessly in the canopy before launching lightning-fast attacks on sloths, monkeys, or other arboreal mammals. Their feather patterns provide camouflage that makes them virtually invisible against the dappled light and shadows of the forest canopy, while their powerful talons can exert crushing pressure that instantly subdues prey much larger than themselves. Snake predators in rainforests have evolved diverse hunting strategies that exploit different aspects of the forest environment. Some species are ambush predators that remain perfectly still for days or weeks, waiting for prey to come within striking distance. Others are active hunters that search through the forest using chemical cues to track potential victims. The emerald tree boa represents the pinnacle of arboreal snake evolution, with heat-sensing pits that can detect the body warmth of mammals and birds, prehensile tails that provide secure anchoring while striking, and perfectly cryptic coloration that makes them invisible among forest foliage. The prey species of rainforests have evolved equally sophisticated strategies for detecting and evading predators. Many arboreal mammals possess enhanced sensory capabilities that allow them to detect approaching threats before they become visible. Spider monkeys use sentinel behavior, with individuals taking turns watching for predators while others feed, creating early warning systems that benefit entire social groups. Their alarm calls are often species-specific, providing detailed information about the type of threat and its location to help group members select appropriate escape responses. The escape strategies employed by rainforest prey species demonstrate remarkable innovation in three-dimensional movement. Many small mammals and birds have evolved the ability to perform controlled falls, dropping from high branches and using specialized body positioning to land safely on vegetation below. Some species can even glide short distances using flaps of skin or specialized feather arrangements that transform a predator-induced fall into a controlled escape maneuver. Mixed species associations provide another layer of predator detection and avoidance in rainforest environments. Many bird species participate in mixed species flocks, where different species contribute different types of vigilance and predator detection capabilities. Some species excel at detecting aerial predators like hawks and eagles, while others are more sensitive to terrestrial threats like snakes and small carnivores. The collective vigilance of these mixed flocks provides superior predator detection compared to what any single species could achieve alone. The temporal dimension of predator-prey relationships adds complexity to these interactions, as many predators and prey species have evolved different activity patterns that minimize direct encounters. Nocturnal predators like owls and bats hunt prey species that are active during darkness, while diurnal predators focus on day-active species. However, the transition periods at dawn and dusk often create windows of vulnerability when both diurnal and nocturnal species may be active simultaneously, leading to some of the most intense predation events in rainforest ecosystems. Specialized predators play crucial roles in maintaining ecosystem balance by preventing any single prey species from becoming overly abundant. Army ants represent one of the most effective predatory forces in rainforest ecosystems, with colonies containing millions of individuals that can overwhelm and consume virtually any small animal they encounter. Their periodic raids create moving zones of predation pressure that prevent ground-dwelling invertebrates from becoming too concentrated in any particular area. The relationship between predators and herbivorous prey has indirect but profound effects on plant communities. By controlling herbivore populations, predators help prevent overgrazing and maintain the diversity of plant species that might otherwise be eliminated by excessive herbivory. Large predators like jaguars help control populations of herbivorous mammals like peccaries and deer, whose browsing and trampling can significantly affect forest regeneration and plant community composition. The evolutionary arms race between predators and prey drives continuous innovation in both groups, leading to increasingly sophisticated adaptations for hunting and escape. Venomous snakes have evolved increasingly potent toxins, while their prey species have evolved enhanced resistance to these toxins. Similarly, as predators develop better sensory capabilities for locating prey species evolve improved camouflage and concealment strategies. Predator specialization often leads to coevolutionary relationships, where predators become increasingly adapted to hunting specific types of prey, while those prey species evolve specific defenses against their particular predators. These specialized relationships can become so intimate that the predator and prey species influence each other's evolution over thousands of generations, creating matched sets of adaptations that reflect their shared evolutionary history. The chemical dimension of predator-prey interactions involves complex systems of chemical communication and chemical defense. Many prey species produce alarm pheromones when attacked that warn nearby individuals of danger, while some predators use chemical camouflage to mask their scent from chemically sensitive prey. Plants contribute to these chemical interactions by producing compounds that can either attract predators to areas where herbivorous pests are feeding or repel predators that might threaten mutualistic animal partners. Size-selective predation creates important ecological effects by influencing the age and size structure of prey populations. Predators that preferentially target young or small individuals may have different ecological impacts than those that focus on large adults, affecting not only population dynamics, but also the genetic composition of prey populations over time. These selective pressures can influence the evolution of prey life history strategies, affecting everything from reproductive timing to growth rates. The indirect effects of predation extend far beyond the immediate predator-prey relationship to influence entire ecological communities. The presence of predators can alter the foraging behavior of herbivores, causing them to avoid certain areas or change their feeding patterns in ways that affect plant communities. These behavioral modifications can be as important as direct predation in shaping ecosystem structure and function. Keystone predators have disproportionately large effects on ecosystem structure relative to their abundance, often maintaining diversity by preventing dominant species from monopolizing resources. The removal of keystone predators can lead to cascading ecological changes that dramatically alter entire ecosystems, demonstrating the crucial role that predation plays in maintaining ecological balance. The spatial distribution of predation pressure creates a mosaic of areas with different levels of risk throughout the forest, influencing where prey species feed, reproduce, and establish territories. Areas near water sources, fruit trees, or other concentrated resources may experience higher predation pressure, while remote areas might serve as refuges where prey species can engage in vulnerable activities like mating or raising young. Seasonal variation in predator-prey interactions reflects changes in resource availability, breeding cycles, and environmental conditions. During periods of fruit abundance, increased prey activity may lead to higher predation rates, while seasonal migrations or breeding aggregations can create temporary concentrations of both predators and prey that intensify these interactions. The role of scavenging in rainforest ecosystems adds another dimension to predator-prey relationships, with many species opportunistically feeding on carrion when it becomes available. This scavenging behavior helps ensure that energy and nutrients from deceased animals are rapidly recycled back into the ecosystem rather than being lost to decomposition. Human activities can disrupt natural predator-prey relationships through habitat fragmentation, hunting pressure, or the introduction of non-native species. These disruptions can lead to cascading ecological changes that affect entire ecosystems, highlighting the importance of maintaining intact predator-prey relationships for ecosystem health. The study of predator-prey relationships in rainforests continues to reveal new levels of complexity and sophistication in these interactions. Advanced tracking technologies and long-term ecological studies are providing insights into the subtle ways that predation influences ecosystem dynamics and the remarkable adaptations that allow both predators and prey to coexist in these complex environments. The balance between predators and prey in rainforest ecosystems demonstrates how apparent conflict can actually serve cooperative functions at the ecosystem level, maintaining diversity, preventing monopolization of resources, and driving the evolutionary innovation that creates the remarkable biodiversity that characterizes these environments, setting the stage for understanding how isolated forest patches serve as special laboratories for evolutionary experimentation. Within the vast expanse of tropical rainforests lie countless smaller worlds, isolated patches of habitat that function as evolutionary laboratories, where unique forms of life develop in relative isolation from their surrounding ecosystems. These biological islands, whether separated by water, elevation, geology, or distance, create conditions that foster some of nature's most remarkable experiments in adaptation and speciation. From the cloud forests that crown remote mountain peaks to the forest fragments scattered across floodplains, these isolated environments serve as crucibles of evolutionary change, nurturing innovations that contribute to the overall diversity and resilience of rainforest ecosystems. The concept of biological islands extends far beyond the traditional oceanic islands that inspired Darwin's observations of evolutionary processes. In rainforest landscapes, isolation can occur at multiple scales and through various mechanisms, creating opportunities for populations to diverge evolutionarily even within seemingly continuous forest environments. A mountaintop separated from other peaks by intervening valleys, a forest patch surrounded by grassland or wetland, or even a single large tree that hosts unique epiphyte communities can function as an evolutionary island where distinct biological processes unfold. Mountain peaks, rising above the general forest canopy, create some of the most dramatic biological islands within rainforest regions. These elevated environments experience different climatic conditions than the surrounding lowland forests, with cooler temperatures, increased precipitation, and often persistent cloud cover that creates unique growing conditions. The cloud forests that develop on these peaks support plant and animal communities that may share few species with the lowland forests just a few hundred meters below in elevation. The isolation of mountain peaks often dates back millions of years, providing sufficient time for endemic species to evolve in response to the unique environmental conditions found at high elevations. Many mountain peaks support plant species found nowhere else on Earth, having evolved in isolation from their lowland relatives over geological timescales. These endemic species often show remarkable adaptations to the cooler, mistier conditions of their mountain homes, with modifications in leaf structure, growth patterns, and reproductive strategies that reflect their specialized environment. The vertical isolation of mountain forests creates gradients of environmental conditions that can support distinct biological communities at different elevations along the same mountain slope. These elevational zones may be separated by relatively small differences in altitude, yet each supports different assemblages of species adapted to specific combinations of temperature, moisture, and atmospheric pressure. The transition zones between these elevational bands often represent areas of particularly high biodiversity, where species from adjacent zones intermingle and potentially hybridize. Forest fragments separated by rivers, wetlands, or other barriers create another type of biological island within rainforest landscapes. These patches may support populations that become genetically isolated from their counterparts in other fragments, even when the separating distances are relatively small. Over time, these isolated populations may diverge through genetic drift, natural selection, or founder effects, potentially leading to the evolution of distinct subspecies or even new species. The size of forest fragments plays a crucial role in determining their capacity to support evolutionary processes and maintain viable populations of different species. Larger fragments can support larger populations that are less vulnerable to genetic bottlenecks and random extinctions, while smaller fragments may experience more rapid evolutionary change due to stronger genetic drift effects. The balance between population size and isolation creates different evolutionary pressures that can lead to divergent outcomes, even in fragments that experience similar environmental conditions. Edge effects around forest fragments create gradients of environmental conditions that can promote evolutionary divergence within species populations. Individuals living near fragment edges experience different light, temperature, and humidity conditions than those in fragment interiors, potentially leading to the evolution of distinct ecotypes adapted to edge versus interior environments. These microevolutionary processes can contribute to overall genetic diversity within species while potentially setting the stage for future speciation events. Geological diversity within rainforest regions creates natural mosaics of habitat types that function as biological islands separated by substrate rather than geographic barriers. Areas with different soil types, rock formations, or drainage patterns may support distinct plant communities that, over time, can promote the evolution of specialized species adapted to particular geological conditions. Serpentine soils, limestone karst formations, or areas with unusual mineral compositions often support endemic plant species that have evolved tolerance for these challenging growing conditions. Wetland areas within rainforest landscapes create aquatic and semi-aquatic islands that support communities quite different from those of the surrounding terrestrial forest. These wetland islands may be connected to other aquatic habitats during high water periods, but become isolated during dry seasons, creating opportunities for aquatic species to undergo evolutionary divergence in temporary isolation. The seasonal connectivity and isolation of wetland habitats creates complex patterns of gene flow that can promote both local adaptation and the maintenance of genetic diversity. Epiphyte communities living on individual trees or groups of trees represent biological islands at a much smaller spatial scale, yet they can support remarkable diversity and endemic species. Large, long-lived trees may accumulate epiphyte communities over decades or centuries, with different tree species or individual trees supporting distinct assemblages of plants and associated animals. The isolation of epiphyte communities between trees can promote local adaptation and potentially contribute to the evolution of host-specific epiphyte species. The temporal dimension of biological islands adds another layer of complexity to these evolutionary processes. Habitats that are currently isolated may have been connected in the past, while areas that are currently connected may become isolated through future environmental changes. These temporal patterns of connection and isolation create opportunities for populations to exchange genes during periods of connectivity while undergoing independent evolution during periods of isolation. Climate refugia represent a special type of biological island that becomes particularly important during periods of environmental change. Areas that maintain suitable habitat conditions during times when surrounding regions become inhospitable can serve as refugia, where species persist and continue evolving while their relatives in other areas face extinction. These refugia often become centers of endemism, where species that survived environmental changes in isolation subsequently diversify to exploit local opportunities. The role of biological islands in maintaining genetic diversity extends beyond their function as sites of speciation to include their importance as repositories of genetic variation within species. Isolated populations often maintain genetic variants that may be lost from larger, more connected populations due to genetic homogenization. This genetic diversity, stored in isolated populations, can be crucial for species' ability to adapt to future environmental changes. Seed dispersal patterns strongly influence the degree of isolation experienced by plant populations on biological islands. Species dispersed by animals may maintain genetic connections between isolated habitats through occasional long-distance dispersal events, while species with limited dispersal capabilities may experience stronger isolation effects. The dispersal abilities of different species create different patterns of genetic connectivity that influence evolutionary trajectories in island environments. Pollination networks also affect the evolutionary dynamics of plant species on biological islands. Plants that rely on specialized pollinators may experience stronger isolation effects than those pollinated by more mobile generalist species. The breakdown of pollination relationships on small islands can lead to reproductive isolation and potentially drive the evolution of self-pollination or other alternative reproductive strategies. The conservation implications of biological islands within rainforest landscapes highlight the importance of maintaining connectivity between habitat patches while preserving the isolation that promotes evolutionary innovation. Corridor establishment can help maintain gene flow between populations while still allowing for the local adaptation that contributes to overall biodiversity. Understanding the balance between connectivity and isolation is crucial for designing conservation strategies that maintain both current biodiversity and evolutionary potential. Human activities can both create and destroy biological islands within rainforest landscapes. Habitat fragmentation creates new islands while often reducing the size of existing ones, potentially altering evolutionary trajectories in ways that may reduce rather than enhance biodiversity. However, restoration activities can also create new habitat islands and reestablish connections between previously isolated areas. The study of biological islands within rainforest environments continues to reveal new insights into evolutionary processes and the factors that promote or constrain biodiversity development. Long-term monitoring of island populations provides opportunities to observe evolutionary change in real time, while molecular genetic techniques allow researchers to track gene flow patterns and evolutionary relationships between populations. These islands of innovation demonstrate how spatial heterogeneity and isolation can nurture evolutionary creativity, contributing to the remarkable diversity that characterizes rainforest ecosystems. The mosaic of connected and isolated habitats creates opportunities for both local specialization and broad adaptation, maintaining the evolutionary flexibility that allows rainforest communities to persist and adapt over long time periods, preparing us to explore how these diverse communities maintain their health and resilience through natural healing processes. The rainforest's capacity for healing represents one of nature's most remarkable demonstrations of resilience and renewal. When hurricanes topple ancient giants, when landslides scar hillsides, or when human activities fragment pristine landscapes, the forest responds not with surrender, but with an orchestrated recovery that draws upon millions of years of evolutionary experience in overcoming disturbance. This healing process reveals the rainforest's deepest nurturing qualities as the ecosystem mobilizes its vast biological resources to repair damage, restore function, and often emerge stronger and more diverse than before. Natural disturbances have shaped rainforest ecosystems throughout their evolutionary history, creating selection pressures that have favored species and communities capable of rapid recovery. Hurricanes, which may strike tropical forests with devastating force, create patterns of destruction that initially appear catastrophic, but ultimately provide opportunities for forest renewal. The powerful winds that topple canopy giants simultaneously open the forest floor to sunlight, triggering seed germination and stimulating the growth of plants that have waited years or decades for such opportunities. The immediate aftermath of a hurricane presents a dramatically altered landscape where the forest's carefully structured layers have been disrupted. Canopy trees that once formed a continuous green ceiling now lie scattered across the forest floor, their massive trunks creating a complex topography of fallen logs and exposed soil. Yet within days of such disturbances, the forest's healing mechanisms begin to activate. Seeds that have remained dormant in the soil seed bank suddenly find themselves exposed to the light and moisture conditions they require for germination. Pioneer species represent the rainforest's first responders, specially evolved to colonize disturbed areas and begin the process of ecological succession that will eventually restore forest structure. These species possess characteristics that enable rapid growth and reproduction in the harsh conditions that follow disturbances. Fast-growing trees like Cecropia can establish themselves quickly in disturbed areas, their light seeds capable of traveling considerable distances to colonize newly opened sites. The seeds of pioneer species often possess remarkable longevity, remaining viable in the soil for years or even decades, while waiting for disturbance events to create suitable conditions for germination. This seed bank strategy ensures that pioneer species are ready to respond immediately when opportunities arise, rather than having to disperse into disturbed areas from distant sources. The diversity of pioneer species, each adapted to slightly different post-disturbance conditions, provides multiple pathways for forest recovery. Pioneer trees serve crucial functions in preparing disturbed sites for the eventual return of mature forest species. Their rapid growth quickly reestablishes a canopy layer that moderates temperature and humidity conditions at ground level, creating microclimates more suitable for the establishment of later successional species. The leaf litter, produced by pioneer trees, begins to rebuild the organic layer that is essential for nutrient cycling and soil development. The root systems of pioneer species help stabilize soil and prevent erosion on disturbed sites, particularly important on slopes where landslides or other disturbances have exposed mineral soil. These pioneer roots also begin to reestablish the complex networks of mycorrhizal associations that are essential for efficient nutrient cycling in tropical forests. The rapid biomass accumulation of pioneer vegetation helps capture and retain nutrients that might otherwise be lost from the system through leaching. As pioneer communities mature, they create conditions that favor the establishment of intermediate successional species. The shade cast by pioneer trees eliminates the harsh light conditions that initially favored fast-growing species, while the accumulation of organic matter and the moderation of microclimate conditions enable shade-tolerant species to establish themselves. This transition marks the beginning of a gradual shift toward the complex community structure that characterizes mature rainforest ecosystems. The process of forest succession following disturbance is not simply a linear progression, but involves complex interactions between multiple species and environmental factors. Different pathways of succession may develop depending on the nature of the initial disturbance, the availability of propagule sources, and local environmental conditions. Some areas may develop into communities dominated by different species than were present before the disturbance, while others may eventually return to conditions very similar to the original forest. The role of animals in forest recovery processes extends far beyond their function as seed dispersers, though this remains crucial for reestablishing plant diversity in disturbed areas. Birds and bats that visit recovering forests bring seeds from intact forest areas, introducing species that might not otherwise colonize disturbed sites. The activities of these animal visitors also help reestablish the complex ecological interactions that characterize mature forest communities. Frugivorous birds play particularly important roles in forest recovery by consuming fruits from pioneer trees and depositing seeds throughout recovering areas. The movements of these birds create a continuous rain of seeds that helps accelerate the diversification of plant communities in disturbed sites. Different bird species have different flight patterns and habitat preferences, creating diverse patterns of seed deposition that contribute to the heterogeneity of recovering forests. The recovery of animal communities in disturbed forests often lags behind plant community recovery, as many forest animals require specific habitat characteristics that may take years or decades to develop. However, the gradual return of animal diversity helps accelerate forest recovery by reestablishing pollination networks, seed dispersal relationships, and other ecological interactions that are essential for ecosystem function. Insect communities typically recover more rapidly than vertebrate communities following forest disturbance, as many insect species have short generation times and high dispersal capabilities. The establishment of insect communities is crucial for forest recovery because these organisms provide essential services, including pollination, seed dispersal, and decomposition that support the recovery of other components of the ecosystem. The recovery of epiphyte communities represents one of the most challenging aspects of forest healing, as these aerial plants must wait for suitable host trees to develop and create the microclimate conditions required for epiphyte establishment. The process of epiphyte recolonization may take decades or centuries, depending on the availability of propagule sources and the development of appropriate host tree communities. Large-scale disturbances can provide opportunities for genetic mixing and evolutionary change within forest communities. When forest fragments become reconnected through natural recovery processes, previously isolated populations may experience gene flow that reduces inbreeding and increases genetic diversity. This genetic exchange can enhance the adaptive potential of recovering populations and contribute to long-term ecosystem resilience. The healing process in rainforests demonstrates remarkable flexibility in response to different types and scales of disturbance. Small-scale disturbances like individual tree falls may be filled by lateral growth from adjacent trees and rapid colonization by understory species already present in the area. Larger disturbances may require more extensive succession processes involving multiple stages of community development. Human-caused disturbances often present greater challenges for forest recovery than natural disturbances, as they may involve soil degradation, chemical contamination, or ongoing disturbance pressures that impede natural healing processes. However, the fundamental recovery mechanisms that rainforests have evolved remain functional even in human-disturbed landscapes, though recovery may be slower and follow different trajectories than natural succession. The restoration of degraded rainforest areas often benefits from human intervention that works with natural recovery processes rather than attempting to replace them. Techniques like nucleation, where small patches of native vegetation are established to serve as sources of seeds and habitat for recolonizing species, can accelerate natural recovery processes by providing the biological infrastructure needed for ecosystem healing. Understanding forest recovery processes has important implications for conservation and restoration strategies. Protecting intact forest areas that can serve as sources of seeds and recolonizing species is crucial for landscape-scale recovery processes. Maintaining connectivity between forest fragments helps ensure that recovering areas have access to the biological diversity needed for complete ecosystem restoration. The temporal scales of forest healing processes highlight the importance of long-term conservation planning and management. While pioneer communities may establish within years of disturbance events, the recovery of mature forest characteristics may require decades or centuries, The full restoration of complex ecological interactions and specialized species may take even longer, emphasizing the importance of preventing disturbances when possible. Climate change adds new dimensions to forest recovery processes by altering the environmental conditions under which succession occurs. Changes in temperature and precipitation patterns may favor different species during recovery than would have been successful in the past, potentially leading to the development of novel forest communities adapted to changing climatic conditions. The study of forest recovery processes continues to reveal new insights into the mechanisms that enable ecosystem resilience and the factors that promote or constrain healing processes. Long-term monitoring of recovering forests provides valuable information about succession trajectories and the effectiveness of different restoration approaches. These healing powers of the rainforest demonstrate the remarkable capacity of complex ecosystems to repair themselves when provided with time and appropriate conditions, revealing how the nurturing qualities that support everyday forest life extend to processes of recovery and renewal that ensure the persistence of these vital ecosystems across time, setting the stage for understanding how forests serve as living laboratories that continue to teach us about the principles of sustainable life. As our journey through the rainforest draws to a close, we find ourselves not at an ending, but at a beginning, standing before one of science's greatest ongoing adventures. The rainforest continues to function as Earth's most productive laboratory, where new discoveries emerge with each passing year and where our understanding of life itself continues to evolve. These forests nurture not only the countless species that call them home, but also human knowledge, providing insights that reshape our understanding of ecology, evolution, medicine, and sustainability. The lessons learned within these green cathedrals extend far beyond their boundaries, offering guidance for how humanity might live more harmoniously with the natural world. The pace of species discovery in rainforest environments remains staggering, with scientists estimating that we have identified less than half of the species that exist in tropical forests worldwide. Each expedition into remote forest areas yields new species, from microscopic fungi to colorful birds, from tiny insects to previously unknown plants. The technological advances that enable these discoveries continue to reveal the extraordinary depth of rainforest biodiversity, with DNA sequencing techniques uncovering cryptic species that appear identical to the naked eye, but represent distinct evolutionary lineages. Recent discoveries have revolutionized our understanding of the complexity of rainforest ecosystems. The discovery of canopy soils, rich organic layers that accumulate in the crowns of large trees, revealed entire aerial ecosystems that were previously unknown to science. These suspended gardens support communities of plants and animals that never touch the ground, demonstrating how rainforests create habitat in dimensions that researchers had never fully appreciated. The exploration of rainforest canopies through techniques like canopy cranes and tree climbing has opened new frontiers in ecological research, revealing that many rainforest species spend their entire lives in the aerial environment above our heads. These canopy studies have uncovered new forms of plant-animal interactions, previously unknown nutrient cycling pathways, and evidence of complex chemical communication networks that operate throughout the three-dimensional space of the forest. Underground explorations have been equally revelatory, with researchers discovering that the root systems and mycorrhizal networks of rainforest trees are far more complex and extensive than previously imagined. The realization that trees can share resources through underground fungal networks has fundamentally changed how we understand forest ecology, revealing cooperation where we once saw only competition and demonstrating the interconnected nature of forest communities. The study of rainforest microbial communities continues to yield discoveries that challenge our understanding of life's diversity and complexity. Single soil samples from tropical forests can contain thousands of bacterial and fungal species, many of which are unknown to science and possess unique biochemical capabilities. These microorganisms play crucial roles in nutrient cycling, plant health, and ecosystem function, yet we are only beginning to understand their importance and potential applications. Rainforest research has provided fundamental insights into ecological principles that apply far beyond tropical environments. The concept of keystone species, the understanding of trophic cascades, and the recognition of the importance of habitat connectivity all emerged from or were refined through rainforest studies. These concepts now inform conservation and management strategies worldwide, demonstrating how rainforest research nurtures understanding that benefits ecosystems globally. The study of succession and forest recovery in tropical environments has provided valuable insights into ecological resilience and restoration. Understanding how rainforests heal themselves after disturbance has informed restoration efforts in degraded landscapes worldwide, while studies of forest fragmentation have revealed the importance of maintaining habitat connectivity for ecosystem health. Climate research conducted in rainforest environments has been crucial for understanding global climate patterns and predicting future climate change impacts. Rainforests play essential roles in global carbon cycling, water cycle regulation, and weather pattern generation. Long-term monitoring stations in tropical forests provide critical data about how these ecosystems respond to changing environmental conditions and how those responses might affect global climate systems. The pharmaceutical potential of rainforest biodiversity continues to drive medical research and drug discovery efforts. Many of our most important medicines originated from rainforest plants, and ongoing research suggests that tropical forests may contain treatments for diseases that currently have no cure. The complex chemical interactions between rainforest plants and their environment have evolved sophisticated defense compounds that may hold keys to new medical treatments. Biomimicry research in rainforest environments has inspired technological innovations that benefit human society. The adhesive properties of gecko feet, the water-repelling surfaces of lotus leaves, and the efficient structures of bird wings have all inspired engineering applications. The three-dimensional architecture of rainforests provides models for sustainable building design and urban planning that maximize space utilization while minimizing resource consumption. The study of rainforest agriculture, particularly the traditional farming methods of indigenous peoples, has provided insights into sustainable food production systems. Agroforestry techniques that mimic natural forest structure and function offer alternatives to conventional agriculture that can produce food while maintaining biodiversity and ecosystem services. These approaches demonstrate how human activities can work with, rather than against, natural ecological processes. Rainforest research continues to reveal the importance of biodiversity for ecosystem stability and resilience. Studies showing how species diversity affects ecosystem productivity, disease resistance, and recovery from disturbance have fundamental implications for conservation biology and environmental management. The insurance hypothesis, which suggests that biodiversity provides insurance against environmental uncertainty, emerged from rainforest research and now influences conservation strategies worldwide. The economic value of rainforest ecosystem services has become increasingly recognized through detailed scientific studies of forest functions. Research demonstrating the monetary value of services like carbon storage, water cycle regulation, and biodiversity maintenance has provided economic arguments for forest conservation that complement ethical and aesthetic justifications for protection. Educational programs based in rainforest environments continue to nurture environmental awareness and scientific understanding. Field stations throughout the tropics serve as living classrooms where students and researchers can observe ecological principles in action and develop deeper appreciation for the complexity and importance of natural systems. These educational experiences often inspire lifelong commitments to environmental science and conservation. The role of indigenous knowledge in rainforest research highlights the importance of traditional ecological understanding in modern scientific investigation. Indigenous peoples possess detailed knowledge of forest ecology gained through thousands of years of intimate interaction with these environments. Collaborative research programs that combine traditional knowledge with modern scientific methods often yield insights that neither approach could achieve alone. Technological advances continue to expand the possibilities for rainforest research. Remote sensing technologies allow scientists to monitor forest changes across vast areas and through time, while automated monitoring systems can collect continuous data on forest conditions. Genetic sequencing techniques enable researchers to study microbial communities and trace evolutionary relationships with unprecedented precision. The conservation implications of ongoing rainforest research emphasize the urgent need to protect these ecosystems before their secrets are lost forever. Each species that goes extinct takes with it potentially valuable genetic information and ecological relationships that might have provided crucial insights for science and medicine. The race between discovery and destruction makes rainforest conservation a scientific as well as an environmental imperative. International research collaborations in rainforest science demonstrate how these ecosystems nurture global cooperation and understanding. Scientists from around the world work together in tropical forests, sharing knowledge and resources to address questions that transcend national boundaries. These collaborations model the kind of international cooperation that will be necessary to address global environmental challenges. The inspiration that rainforests provide extends beyond scientific research to influence art, literature, and philosophy. The complexity, beauty, and mystery of these ecosystems continue to capture human imagination and challenge us to think more deeply about our relationship with the natural world. The sense of wonder and humility that rainforests inspire may be as important as their scientific contributions in nurturing more sustainable human attitudes toward nature. Future research directions in rainforest science promise to reveal even more about these remarkable ecosystems and their importance for global environmental health. Studies of climate change impacts, investigations of ecosystem services, exploration of microbial diversity, and development of sustainable use strategies all offer potential for discoveries that could transform our understanding of natural systems and our relationship with them. The global influence of rainforest research extends to policy and decision-making processes at all levels of government and society. Scientific understanding of rainforest ecology and ecosystem services informs international agreements on climate change, biodiversity conservation, and sustainable development. The knowledge generated in rainforest laboratories influences how we think about urban planning, agriculture, medicine, and technology. As we reflect on our journey through the rainforest's many dimensions, from its architectural complexity to its healing powers, we see how these ecosystems embody principles of sustainability, resilience, and interconnection that offer guidance for human society. The rainforest's capacity to nurture life in its countless forms extends to its role in nurturing human understanding and inspiring more thoughtful approaches to living on Earth. The living laboratory of the rainforest continues its patient work of innovation and discovery, offering lessons in cooperation, efficiency, and adaptation that humanity desperately needs to learn. In protecting these forests, we protect not only the countless species they contain, but also our opportunities to understand and learn from one of nature's greatest achievements, ensuring that future generations can continue to discover the secrets these remarkable ecosystems hold and apply their lessons to creating a more sustainable world.